Yeah, if you like. I had friends who used to work in Birmingham. Right now in South Wales University as a lecturer in psychology, 1 hour a day. Really? Okay, I started also with yeah. Otherwise, we could work together. It's really Before that, I knew nothing about the spinal cord or pain. I only started when I started working started my new career until now. Personally, nothing about it. Only for the last couple of years, I started more different types of So when you talk, you yes, it's tough. Exactly. Then we can remove my aim also will protect the stage because yes. With different noise in the network, actually transfer out the rather reproducibility increase. Also, we have kind of simple to achieve for our subjects. Whether that means we use that basically, we're working on that. We're full factor of the results. Now we have like 32 we can for PCA, we can have 5, 3, 10 values. For ICA, we can have a and what ends is like ultimate, or is it not? We cannot. That's what further from the mistake of the degree of freedom. And I don't like whether this really is like at least now I feel like ICA is very yeah, now my question is whether we have the other if you have that, but how much that we benefit? Yeah. Actually, John and I, we developed that software. So we inducted very straightforward. It's great.
Good morning, everybody. Let's just start the second day. There is coffee in the back, serve yourself. Today we have a little bit of milk and water. Then we have chairs, Marcello and Sarah. I leave to you the introduction of the speaker.
Yeah, so the first speaker is Claudia Gandini, which is professor here at UCL and in idea of MR physics. And the first talk is about spinal cord as part of the central nervous system, so advances in the brain and spine.
Okay, thank you very much for inviting me to speak. I can't remember why or how I choose this start. No, I do remember, so I hope that I'll defend the idea that now that we are becoming quite good at acquiring spinal cord data, we should move and try to plan our protocols to acquire always brain and spine in a coherent manner. So I will try to address the issue by, I can't see the point. Okay. Anyway, by starting arguing that in multiple sclerosis it's obvious that is what we should be doing all the time. But there is also an argument that in other diseases where it's less obvious that we need to look at the brain and spine, we should really look into it. And then I would discuss the limitations of the old approaches. So what are we doing at the moment with our protocols? And then, in my opinion, I will tell you later again, but it's because we optimize protocol independently from each other and because of the longer position time. And then some solutions, and showing a couple of studies that we are currently doing that actually are addressing the points. So normally when I show this picture is to show that the brain is very large and the spinal cord is very small, and it's a challenge for acquisition methods. But in this case, I want also to say that there is a lot of information that goes and comes to the brain from the spinal cord. So this little piece of neural tissue that we have in the spinal cord is essential for a feedback from all the sensory system and for all our motor actions. And our motor is also linked to our cognitions and our functioning. So I think that the spinal cord is very relevant in many, many situations. And this is what we should be doing, looking at spinal cord and brain. This is the pictures from Julien. And I think that this is still the case, that for acquiring something like this, it takes a long time and a lot of effort to put it all together in this way. But this is where we should aim. This is what we should be doing. So we all know that in MS, we've got lesions, and lesions are visible in the brain and in the spinal cord. And clinically now, the spinal cord lesions are also included in the diagnostic criteria. But if we go and talk to Nikos Evangelou in Nottingham, he would tell you that the spinal cord is actually one of the most relevant parts of the central nervous system affected in multiple sclerosis. But not only the spinal cord, because if you look at the cerebellar cortex as well as other part of the brain cortex, neobrain cortex, you have a lot of lesions also in the cerebrum. So we can't just look at the cord, but we should neither just look at the cortex and the white matter in the brain. So we've been very good at, unless I missed quite a lot of the literature, at studying white matter lesion, studying normal appearing white matter, gray matter, hyperintense lesion, hypointense lesions, and you call it, you name it. No, you can find papers that look at the brain and quantitative metrics of the brain in MS. And sometimes in some studies, we also look at the spinal cord in a quantitative manner. So we do look at microstructure indices in the spinal cord. Normally in the cervical spinal cord, although some work is now being done in the lumbosacral part of the brain, of the spine, sorry. And we've been now developing as well techniques that allow us to extract white matter and gray matter in the cord, lesion in the cord. So we are publishing and looking at the cord as itself with quantitative metrics. But how many times have we published something that shows spinal cord and brain together in the same paper to look at a metric that investigates the property of the central nervous system coherently between the two of them? I didn't do I didn't spend a long time searching for them, but they don't jump out very easily. Now, Sarah here published this paper of the brain and spinal cord using NODI. NODI is a multi-shell method that extracts diffusion method that extracts microstructure tissue properties like neurite density and orientation dispersion. And in this paper, Sarah has shown that there are alterations in the cord and in the brain and in the cord and are actually specific to certain tracts. And also she showed that when you're looking at correlation with DDSS, so with clinical disability, the spinal cord, the neural density in the spinal cord white matter is really important. Now, if I may, Sarah, this paper, although we acquired the data in a coherent manner between the brain and the spinal cord, we published it with brain and spinal cord because we were so late in publishing and in analyzing the data that we thought that just the brain was not interesting enough. So we put it together and we did it, and it's actually, the data was acquired in a coherent manner, but it was a bit of a post-analysis thinking of looking at the spinal cord and the brain in the same paper, looking at the spine and the brain as a coherent part of the central nervous system. But this is what we should do. And we could do it because with hindsight, we applied the brain protocol to the spine, but you know, we should do this more often and you know, in all our studies. Now let's look at the spinal cord injury, where we've got alteration of the spinal cord, but the brain in theory should be healthy. Now, Patrick Freund, a few years ago, when he was here in London, then he went back to Zurich, did a study looking at individual tracts. This was still 1.5 Tesla state, I think. And he showed that we have differences in diffusion tensor metrics in the cord and in specific tracts. And actually, I really like as well this combined DTI and MTR study from Julien, where it showed that in spinal cord injury patients, we have altered metrics, but the MTR was actually specific to the motor or the sensory component of the ASIA score. So the microstructure of the spinal cord above and below the lesion in spinal cord injury chronic patients are able to are sensitive and correlate with specific motor or sensory scores. But what happens to spinal cord injury patients in the brain? So this was Patrick that started insisting on and talking about the neuroaxis. So he looked at the corticospinal tract and showed actually that there were differences in FA in this case, along the corticospinal tract, in the pyramids, in the posterior internal capsule, and in the motor M1 motor area. So the brain of chronic spinal cord patients is altered as well as the spinal cord. And so you could say, okay, it's Wallerian degeneration, so the brain, of course, in spinal cord injury patients, they don't move their bodies and the injury goes back and affects the brain. But in a recent, and also he showed that, of course, there was some correlation with clinical scores of using the FA of the right corticospinal tract. But in a recent paper of acute spinal cord injury patients, what he found was that not only the cord structural matrix were altered, so the width and the thickness of the spinal cord and the spinal cord area, but also there were a lot of brain changes immediate, very, very soon after the spinal cord injury. And especially when you looked at predictive value of the alterations early in the brain and in the spinal cord for how the patient stayed 12 months after, what was the two things that were predictive of patients' outcome were in the spinal cord, the width of the anterior posterior width of so the thickness of the cord basically, but also the cerebellar microstructure changes and volume changes in the cerebellum. So the spinal cord and the brain at very soon, at acute stage, are predictive of what happened to the patient. So it's not only what happens locally, it's also what happens very early in the spinal cord injured neural tissue in the disease that is determining the outcome of the patient. So we shouldn't look in these patients' spinal cord injury just at the spinal cord. Even in that, we should look at the brain. Now, what happens the other way in a brain disease that is just or known as neurodegenerative disease that affects the brain, in particular frontal lobe or temporal lobe, like in Alzheimer? We have clear pathways and clear changes happening in the Alzheimer brain and atrophy of the medial temporal lobe. We asked ourselves what happens in the spinal cord. This is something that I had in mind to do a lot, and finally I found a student in my group in Italy that was agreed to take this on. So with the spinal cord toolbox, we wondered, is the cervical cord atrophy a marker also of disease of degeneration in Alzheimer? And the paper has just been accepted. We have received the proof yesterday, but what I want to show you that what we did was to parcellate the brain and go and get to the thalamus and the amygdala, and of course the hippocampi, as well as white and gray matter of the brain that are key parameters that we use for measuring the Alzheimer's disease, but also segmented the cord, the cervical cord, from the same 3D-T1. And then we went and used some feature ranking. We had a pool of 30 odd features to see, because we had only a few subjects, so we extracted a subset of the subject in an iterative way and went and looked what came out. And the spinal cord area, C2-3, kept coming up together with the hippocampus, the amygdala, and the white and gray matter volume. So the spinal cord volume is up in the features that selects and differentiates patients from healthy subjects. And when you go and do the ROC curve on the remaining subjects of the group, you get quite a high performance, considering that this is a small group and data not required for this purpose, which shows us that then you go in literature, you find as well some interesting piece of work in animal models and one post-mortem work on this, on spinal cord of Alzheimer, but I don't want to go into it. What I want to say is that even if it was just a biomarker of progression, we should not forget that the spinal cord is part of the central nervous system and should be investigated also when we have brain-specific disease. So now that I've done my speech and defended why we should do brain and spinal cord together, we have got a 3 Tesla MRI scanner, has been upgraded. It can do all beautiful things. We can do morphological studies. We can do physiological studies. We can do white matter, microstructure, myelin, and we can do the same in the spinal cord, because we've got protocols normally that can do almost everything that we can do in the brain. We can even do sodium quantification in the spinal cord. Pavna is doing a study in spinal cord injury with sodium quantification. Why are we not doing them together then? So here is my argument. I couldn't think of any other reasons than long acquisition time. So we're not doing it because we can't keep the patient in the scanner for hours and hours and hours. If we want to do myelin, diffusion, atrophy, everything, all at the same time. And these cost money. Well done. Hugo suggested that these would cost a lot of money to do everything all the time. And also, when we do it, why don't we publish together spinal cord and brain results like Sarah did? Because we normally optimize the protocol independently, and we end up with something that actually shows slightly different things. So it's much easier to defend something that is shown in the brain than in the spine. There are other potential factors into it, but I think that these two are the main thing. So in this study, which is the INSPIRE study that we've been doing with Julien, I see John and Dave there, and Becky, I don't see her, but anyway. We've been doing it with Toronto, Montreal, and Zurich. So in Toronto and in Zurich, they've acquired data, and between Montreal and the trial office at UCL, we've analyzed the data. So Becky looked at the cortical spinal tract, putting together data that we acquired on the spinal cord with data that we acquired on the brain. And we selected the tracts as anatomy tells us that they should be put together. And then we went and looked at metrics along the cortical spinal tract in spinal cord injury or spinal cord cervical spondylotic myelopathy, CSM, anyway. So we did this, but this was a post-hoc analysis. So we have acquired diffusion in the cord and diffusion in the brain, optimized or acquired separately, as clinical protocols were suggesting. So for various reasons, the p-value of the spinal cord data was very low, was rather low, was 500. And also, if you look at these two protocols, brain and spinal cord, the brain had, because of time, had a resolution of 2.5 by 2.5 by 2.5 millimeter cubed, while the spinal cord had a 0.8 by 0.8 by 5. So they have a very different acquisition. B factor and resolution are very different, even in volume. So anyway, we decided to downsample the brain in chunks of 5 millimeters so that at least we were looking at the plots along the cortical spinal tract in the same way. So here we've got an example of brain and spinal cord. And here is what happens. So you have the diffusion data in the spinal cord and in the brain behaving quite coherently between the subjects. So these are all the subjects put together between Toronto and Zurich. So two different sites in two different continents. They give quite consistent numbers between them of patients and of health control. The spinal cord is where you find the most differences when you go and do some statistics. But one, you miss a chunk of the cortical spinal tract because the two protocols were not done overlapping. And also you have different metrics probably because of those different B factors that were used, so sensitive to different microstructure. So here we go. So how can we make this better? So we've got two solutions. So my first solution, extend the 3D acquisition to cover the cord. You can do this, I think, when you have 3D techniques. So when you've got a volume of the brain that has a good resolution of 1 by 1 millimeter cube minimum, because in the cord you need the resolution. You can't go with 2.5 or 2 millimeter by 2 millimeter by 2 millimeter resolution. You wouldn't see anything. And it's been shown that for atrophy measure, this can be done. This is indeed what we did for the Alzheimer's study, that we used structural data that was acquired for structure of the brain. And we have looked at the MPM, which is a protocol, again, that uses the 3D acquisition, and that because of the 3D volume, you end up covering the spinal cord as well, a little bit of the cervical spinal cord. And this, Becky again compared the the spinal cord MT parameters, MPM parameters from the brain protocol with those acquired with a dedicated spinal cord protocol. And if you're looking at the whole cord area and you don't want to go and look at white and gray matter, the bit from the whole brain protocol is good enough to give you some measurements. So this is one way, one approach that we can do it. And this has been written up for publications, but this is what we should do with this kind of data. But this wouldn't work with diffusion. And it wouldn't work with diffusion because when you go down to the spinal cord with a massive field of view for the brain, your spinal cord will look horrible for distortions. And also, it would have a resolution that, it would be impossible to make any sense of what you're seeing. Besides, you would have pulsatile, it would be affected by cardiac motion. So the other thing that we did, and these are my last few slides, is to unify the readout. So this, the next study is the MS-QMRI study, the quantitative MRI study. The slides were given to me by Marco, but has been also a team effort in this unified readout, and Francesca that is sitting there, I think worked a lot with Marco and Becky to get this going. So we've got fast acquisition because everything is based on API. We use proper microstructural models. We use the Zoom API on the spinal cord to avoid big distortion and to be able to optimize the resolution. So the protocol of the brain and spine are required separately, but are very much unified. And we are going to do all this study of MS patients longitudinally with different metrics that are, I showed in a couple of slides, are diffusion, microstructure, myelin, and relaxometry. So what we are doing, these two acquisitions for the brain and for the spine are identical, apart from the zoom bit that is just the selection of your voxel of interest, of your region of interest. So everything else is identical. And because of that, you can also unify basically the analysis because they've been acquired with the same API type of readout. And when you get the quantitative parameters out of it, you get quantitative maps that are actually giving you the same things. And we're doing this, we are doing all of these, acquiring therefore relaxometry, so T1, T2 parameters, bound to fraction from Q and T, and then a number of diffusion metrics from DTI in the brain and in the spinal cord in 1 hour, which is very, very good. And it's possible because of the unified readout. The problem of this is, of course, these are some colored matrix maps that I had on the different presentations, but they are the same as the previous very detailed maps. And what you can do, therefore, you can do whatever, you can do gray and white matter analysis, you can do lesions, you can do, but these numbers are comparable, okay? So in conclusion, I think that it's very important that we start designing the protocols in a way that you can look at the brain and spine in a coherent manner for a hypothesis-driven study. And to do this, we should design matrix studies. If you can't do everything, you could at least do something that is coherent between the brain and the spine. And we need to acquire to develop methods that are fast so that we can do all of these in a clinically feasible time. And that we can use 3D sequences extending to the spinal cord, but we can do this only for certain type of applications. And we should try to convince the manufacturers that if we develop something that should be translated to and available to all scanners or all, at least within the same manufacturer, so that things are translatable to clinical studies quicker than what they are actually at the moment. So with this, I'd like to thank my team. Actually, we need to do a new physics team picture. These are all that some people are missing, but thank you very much for the attention.
Claudia, are there any questions?
Actually.
Claudia, thanks for the presentation. It was really interesting as usual. I was particularly intrigued by the studies that you showed on Alzheimer's and on spinal cord injury.
Yeah.
Because results are sort of unexpected.
Yeah.
Do you think that the involvement of the brain during spinal cord injury or the involvement of the spinal cord during Alzheimer's, which is conventionally brain disease, is just a consequence of the main damage, or the main damage has caused general neurodegenerative pathology which affects different areas independently.
So I think in
Do
the
you have
spine,
any preliminary data?
I don't have preliminary data, but I can tell you from the discussions of various papers. So in the spinal cord injury, normally because of the definition of it, especially proper spinal cord injury, where you've got accident or whatever, you have got a healthy tissue, and the injury is at the spinal cord. So the brain is a reaction to the spinal cord. I don't think it's just neurodegeneration. I think it's a real reaction of the brain that tries to stop and take care of the fact that suddenly some major input to the system is not there anymore. So I don't know, the cerebellar changes are there to protect in a way the rest of the brain from massive damage or something. So you stop the communication sooner rather than later or something. I don't know, but I think it's definitely a consequence of what's happening in the spinal cord. In Alzheimer, I don't know. We need many more studies, but the pathological studies show that the amount of amyloid plaques and the damage of the spinal cord is not necessarily correlated with the amount of damage in the brain. In the animal studies, one shows that actually spinal cord is damaged from the myelin before the brain in the animal, but it's an animal model, so it's an animal model. And in the other animal model, I can't remember, but as well, there is something that says that I think it's a generic alteration of what's happening in the neural system that probably then attacks particularly the hippocampus and the brain, because it's what we see. But I don't think that is a consequence. I think it's a general of the central nervous system. And I wonder, if we are going to go and look at everything else, if we find some other stuff as well. After all, the retinal thickness in Alzheimer is altered very early. Maybe a consequence of the brain might be together, not primary, but together at the same time as everything else. So that's my take, but it's one study. So please do, if you've got data, just do it. I've been saying it, we really need to do it. Yeah.
So,
Gabe.
great talk, and you highlight some important issues, especially with Alzheimer's disease and the cord. And we've discussed this before, and some of the literature does suggest that amyloid is deposited in the cord. And if you think about where it's deposited in the brain early, it's in the meningeal vessels, which aren't unique to the brain, right? And so that in itself highlights, as you say, a global central nervous system process rather than something necessarily restricted to those functional areas.
It's just, opening our vision as we can, because we've got the techniques, and try to support mechanistic hypotheses, but not stick to just mechanistic hypotheses that have been there for quite some times. Because I'm not saying that what we've done so far is not right. What I'm saying is that let's just, be open and look at what else we can look at.
I haven't seen the acquisition time of the combined protocol, brain and spine. Can you comment on patient acceptance of that long acquisition?
Well, the acquisition time is 1 hour, which is, for what we're doing, considerably less than what our MS patients are used to. So, Sarah's protocol, the NODI one that does sodium, does all sorts of things, is over 2 hours long. So we are doing all of this in the brain and in the spine in 1 hour. So, the brain protocol is 20 minutes, Marco? Yeah. About 20 minutes. The problem of the length is the fact that the spinal cord, you have to cardiacate. We tried, you can't not cardiacate. We abandoned cardiacated in the brain. Despite I was defending it for a long time, I had to give up, because there are no quantitative differences if you don't cardiacate with the current method. But in the spinal cord, if you don't cardiacate, you get really unreliable results. So patients that have got, not stable heart rate or whatever, take longer, and so you get over an hour and 10 minutes or something. But we do all of that quantitative MT, so with several MT parameters, diffusion, 3-shell diffusion, with the B factor up to 3,000, and we do the relaxometry, so T1 and T2 quantification in the brain and in the spine in 1 hour, or around 1 hour. I think that.
Yeah, I was intrigued. Thanks for a nice talk.
Oh, you don't need to thank.
Motor association with 2-month motor score outcome in the final injury patients.
Yeah.
I'm not correcting that because I was intrigued by the scatter plot. I'm sorry, it's very early on in the talk.
I might. Sorry, I should have got it from there. It was indeed in very early in my talk. This, no, yeah, this.
Yeah. So the left
This.
scatter plot.
This.
Yeah. Is that really associated or?
That's what they published. This is not my paper. I wasn't involved in it. This is the Zurich group.
I don't think they're connected.
I would see two points where
The groups
and align between
of subjects.
where they show multiple.
I think that this was the first paper that they published, and this is last year, so 2019, and I think they had these very early acute patients, and then they called them back after 1 year to do some clinical tests. So these are the clinical tests after 1 year, and this is the width, the anterior posterior width. I agree that's what they claim, that there is an association and of the spinal cord. I think it's more believable the cerebellar outcome than that, but that's there.
And for the joint protocol, how does that tie in with the brain and spinal cord acquisition? Does that tie in with the efforts to standardize?
No, they're not. They're not.
No, that's a different question.
So
a different question about the stuff that you showed.
Yeah.
Joint acquisition for brain
Yeah.
and spinal cord.
Well, at the moment we can. Yeah.
Compared to Julian's standardized protocol.
So the core protocol, we could, one of the shell of the protocol is, so from the point of view of the geometry and whatever, it's similar, and it's a question of number of B factors and whatever. You could do the same things with the protocol that Julien did and doing the same in the brain instead of doing a multi-shell. So you could select the same B factor and acquire in the brain with the same protocol in the brain. So you could add a brain protocol that also, the other thing that I was going to say is that the spinal cord covers, the spinal cord acquisition covers part of the brain acquisition, so that you've got some overlap between one acquisition and the other that allows you to, because you have to have different different voxel size.
Yeah.
So, because you couldn't do the voxel in the brain at millimeter cube level, so you have to have the two different resolutions. But if you acquire and select your volume on the core that overlaps a bit with the volume of the brain, even if it's only for a couple of slices, then it makes it easier to merge the data. So that doesn't make diffusion is easier in a way to standardize, because we just need to pick the right B value for the brain and for the core and try to make it so that you have something that looks at least at the same microstructure. So, yeah.
Okay, thank you, Claudia. And
Okay, thank you very much, Ferran, for the invitation. And so today, what I would like to present are the main results of the studies that applied the spinal cord MRI in multiple sclerosis, which is the field where I work, and with particular regards to disease pathophysiology, so how spinal cord MRI has been used to characterize the different disease pathophysiological processes that are occurring,
And with regards also to disease monitoring, which is a hot topic in these days, so how we can apply spinal cord MRI, especially to monitor disease evolution, to predict the subsequent disease evolution, and which outcomes are still already useful for clinical trials, for including in clinical trials. These are all of topics of discussion, and I will conclude showing you some recent advances that I believe that are promising in this field. So, with regards, of course, to the background, I will go very, very fast because Gabriele already showed us a lot of nice pathological data. So, spinal cord in MS is affected by a lot of different pathophysiological processes, such as demyelination, axonal loss, gliosis, and all of this is really clinically eloquent, so it results in chronic motor sensor and autonomic dysfunction. So it is important to find the best way to image these, to find the imaging correlates of all these pathophysiological processes, and give the strict relationship that there is in the spinal cord between structure and function. It might be a better model than the brain to study the damage of the central nervous system, but of course, because of the reasons that we already heard in these days, it is technically more challenging than the brain imaging, especially with regards to non-conventional MR techniques. But the recent advances have been shown that we can obtain reliable, also non-conventional MRI biomarkers of the spinal cord damage, microstructural damage. So one of the main hallmarks of MS in the spinal cord, as it is the case of the brain, of course, is the presence of hyperintense lesions, T2 hyperintense lesions, which are very frequent. They are also quite specific for MS because are relatively rarely occurring in other neurological conditions. And with regards to the characteristic of lesions that have been traditionally described in conventional MR studies, they have been described to be relatively small and located preferentially in the cervical than in the rest of the cord, even though the remaining cord segments are also not spared. They seem to be preferentially located in the lateral and posterior cord columns, and also present in the central gray matter, as we heard right now. Lesions detection has been done mainly using proton density and T2 weighted scans with the contribution of also short tau inversion recovery scans. But more recently, other sequences have been shown to be useful to detect the presence of lesions, in particular T1 weighted scans, such as the MPRH, has been shown both by study performed at the NIH and also in our lab, showing that there is a nice correspondence between the presence of lesions that you can detect on T2 weighted scans and proton density scans, and lesions that you can detect on high resolution 3D T1. And also PSIR is a very useful sequence in this topic because, for example, in some studies performed here in London, it has been shown that PSIR show a good detection of lesions, and when acquired in the transverse orientation, at least in the upper cervical core section, it is also able to produce some reliable measures of lesion volume in the cervical cord. So that correlates with the disability of patients. Other sequences are also nowadays used to detect lesions in the spinal cord, such as multi-echo sequences. With regards to the use of high field strength, such as 3T or more, Of course, the improvements of magnetic field is not only useful to detect the significant presence of hyperintense abnormalities, but also to show the presence of other types of demyelination, such as the PL and sub-PL demyelination by means of MTR in the outer layers of the cord, as it was shown by the study by Hugh Kearney. And of course, moving to higher field strengths, such as using 7T scanners. You can see much more lesions in the white matter of the spinal cord of MS patients with an increase of up to 52% of the number of lesions that you can detect. So how these lesions, as a consequence of the presence of all these kind of lesions and pathological processes in the spinal cord of MS, one of the first consequences that you can detect on spinal cord MRI images of MS patients is the presence of a significant cord atrophy that was measured in the spinal cord at single cervical cord levels, as Hugo reviewed nicely yesterday, at least on 25 years now. And the study were all coherent in showing a significant decrease of transverse cord area at C2, C3 mainly, which was nicely correlated with clinical disability in these patients. When we used moving to more recent methods proposed for quantification of cervical cord atrophy in the spinal cord, the active surface method that has been illustrated yesterday by Mark, in several studies. One study we performed some time ago in our lab with a very large number of patients, more than 300 patients, showed that there is a well-defined separation between the cord atrophy curves that you can detect in the different MS phenotypes. So the characterization of cord atrophy is helping to distinguish between the various disease clinical phenotypes, and there is also a differential association between cord atrophy and disability across the different types of MS, with no association between these two variables in CIS and benign MS patients, which are characterized with a relatively mild disability, and significant association in the other disease phenotypes. Using the active surface method that allows to produce straightened cord images, it was also possible to improve the characterization of the presence of lesions and the cervical cord atrophy across regionally in the cord, so across the different cord segments and within the transverse cord section. And we did, for example, for instance, these two studies in Milan showing that there was an increased presence, a higher frequency of occurrence of both T2 weighted and T1 hypointense cord lesions in the upper cervical cord segments compared to the lowest cervical cord segments, with a partial but not complete association with the presence of cervical cord atrophy in the different MS phenotypes, so suggesting that there is an association between these two phenomena, lesions and atrophy, but the relationship is not univocal. So we have still more lesions than atrophy. So the location of these phenomena is not completely coherent, so suggesting that there might be later effects of cervical cord lesions, maybe on the subsequent development of cord atrophy. So from the temporal point of view, these two processes are partially independent. So as mentioned now by Hugo, these results were also replicated in the recent study by Julien's group, putting together a lot of patients from more than 20 sites. So also with the spinal cord toolbox, using the spinal cord toolbox was also possible to obtain this nice maps of lesion distribution in the spinal cord that characterized the presence, the different frequency of lesions across the different spinal cord levels, and the differential location of these lesions also within the core section, with the predominant involvement of the posterior and lateral columns. And with regards to studying the lesion, including the lesions, I would like also to mention the study that was recently performed in our lab, in which we not only measured lesion volume in the cervical core section, but also in the thoracic one, and putting together measures of lesion volume in both these cord segments was helping to improve the correlation with the cervical cord atrophy and the clinical disability. So suggesting that even the most of the study concentrated on the cervical cord, also studying the damage, quantifying the damage in other core sections might be useful to improve our correlations to understand the disease physiology. So also, of course, non-conventional MRI techniques have been successfully applied to study spinal cord microstructural damage in multiple sclerosis. So I will focus here mainly on diffusion weighted MRI and magnetization transfer MRI, which are the two techniques that have been mostly applied in this disease, but other techniques are, of course, are welcome in this field for a better description of microstructural damage. The first studies that assessed microstructural damage in the cord using quite relatively simple diffusion models such as the diffusion tensor found that there was the presence of significant microstructural abnormalities in the cord of MS, which was correlated with disability, and almost all the diffusion tensor MR indexes that could be derived in these studies were abnormal and correlated with disability, including also radial diffusivity, which seems to be the best correlator of various measures of sensory motor impairment in this disease. Then I'm happy to present again the work of Sarah's paper and other recent studies that used more advanced diffusion models, such as space imaging, and also again Noddy at Vanderbilt, which was also applied at the Vanderbilt to characterize damage in the spinal cord. And as Claudio and Gabriele already mentioned, these metrics that can be obtained, but more from more advanced diffusion models, can be also useful to describe the damage within the spinal cord, with hopefully a better correlate to the pathophysiological processes, pathological processes that you can see in the spinal cord. Magnetization transfer MRI has been also useful to successfully applied to describe microstructural damage, with closer correlate to the myelination in this case, both in the white matter and in the gray matter of MS patients. There was a significant there were significant abnormalities of MT-derived metrics, which were correlated with the different measures of sensorimotor impairment, and replicating results that were obtained by Hugh Kearney in the previous study. There was also a differential involvement of MTR damage, MTR-related damage in the spinal cord across different levels and within the transverse section, with a more significant decrease of MTR as spinal cord, so in the outer core section, so suggesting again that subpial demyelination is occurring in this structure. So what about the application of this technique, of all these techniques to monitoring the disease evolution in MS? So spinal cord atrophy, so measures of mainly the measures of cross-sectional area at single core level or over more extended portions of the cord, has been applied to monitor disease evolution and predict the conversion from CAS to clinically definite MS, and studies using also methods that measure the cervical cord area at single or cord section between C2 and C3 or C1, C2, showed that there is a significant progression of cord atrophy in all disease phenotype, which seems to be more evident in early primary progressive phases, or also in secondary progressive patients at the beginning of conversion, so when they are close to conversion, but I will discuss this topic later on. And this damage in the spinal cord is able to predict the subsequent disease evolution, at least at medium-term follow-up, while the predictive value of cord damage over extended clinical observation at longer-term follow-ups is still a little bit debated, at least in primary progressive MS. There are some studies that were performed in Milan and London that showing that, of course, when we improve the method of quantification of cord cross-sectional area, we are improving also our reliability of results, especially when applied in the longitudinal settings. And for example, the active surface method was shown by both of these studies as being possibly applied in future clinical trials, because it produces reliable, reasonable sample sizes in different MS phenotypes. And but I will, and in this regards, I think we will hear more from Frederick about the possible quantification, and I will show you also in the last section of one presentation that there are still now improvements in this field with other promising techniques. And I would like also to present the results of this very large study that we recently completed within the Magnus initiative, showing that quantifying cord cross-sectional area between C1 and C7, so covering the entire cervical cord segment, we obtained a significant heterogeneity of atrophy across the different phenotypes, and also a different craniocaudal involvement of the cord across the different phenotypes. So when we analyzed the regional curves of cord atrophy produced by Jim using some spline method applied from our statistician, we were able to find that there was an earlier involvement of the most cranial sections of the cord in the early phases of the disease, and subsequent involvement of the most caudal cord sections in the progressive MS phenotypes. And all these findings were contributing together with the brain atrophy and cord lesions to obtain a nice correlation with disability in our patients. And with regards to the study, we had also a subset of patients for which we had a 1-year follow-up available, and analyzing using the active surface method, with the same method, the 1-year follow-up cord cross-sectional area in these patients, we found that in all phenotypes apart from CAS, there was a significant progression of cord atrophy over time, with the highest rates of atrophy found in early relapsing remitting and in clinically worsened MS. The pattern, the craniocaudal pattern of cord atrophy evolution over time was quite similar to that shown already at baseline, with an earlier involvement of the upper cervical cord at the beginning of the disease, and an extensive, the more extensive involvement of also the lowest cervical cord segments in clinically worsened and in progressive MS. So, and all these results were reflected in these sample size calculations that basically showed that the phenotype in which it might be more worth to investigate, monitor cervical cord atrophy over time for future clinical trials might be early relapsing remitting MS, for which at baseline we have still no cord atrophy, but they are a very high risk of developing cord atrophy in the immediate follow-up. So if we have a treatment that is able to act on these on this metric, on this to prevent this damage, it might be worth to apply it in this phenotype. So with regards to the prognostic value of cord damage, as I told you before, there is still a little bit debate about the value of the cord in predicting the evolution, the subsequent disease evolution at longer term follow-up, because at short-term follow-up there was a quite good correlation between cord atrophy and cord damage at baseline, and clinical disability, clinical status at shortest term follow-up, but this correlation was lost in favor of brain gray matter damage when looking at extended follow-ups, so over 15 years, at least in primary progressive MS, because if we look at relapse onset MS, such as CIS patients, still a 15-year follow-up, we are finding some predictive value of spinal cord lesions, in particular on the evolution to the secondary progressive MS phase. And with regards to this, I would like to mention also the study performed at UCSF, in which there seems to be an accelerated rate in relapsing remitting MS before converting to secondary progressive MS. So cord damage seems to be at least a low point in explaining this conversion, this clinical conversion from one phenotype to another one. There seems to be still a role of the spinal cord on this. And also, with regards to non-conventional MRI, still there is also the possibility to predict subsequent disease evolution, as shown by these two studies, in which cord damage at baseline was able to predict clinical disability over 2.5 years of follow-up, and also was able to predict clinical recovery in a group of MS patients at the onset of a spinal cord relapse. So also assessing microstructural damage seems to be rewarding to characterize damage and predict how patients will behave in the follow-up. So I will conclude with showing some recent advances that I think are promising for monitoring and describing the pathophysiology of MS using spinal cord MRI. of course, gray matter and white matter separation is a very hot topic, so a very good measure of damage in the spinal cord of MS. With regards to the quantification, it has been done for the first time using PSIR sequences of the UCSF and manual quantification, manual delineation, but as shown by the recent challenge launched by Ferran at the SMRN some time ago, it is also possible to segment gray matter automatically in the cervical cord of at least of healthy subjects and in patients not showing T2 visible abnormalities. While there is still some concern about possible quantification of gray matter atrophy when there are some destructive and large MS-related lesions in the spinal cord. And within this field, I found only your preliminary work on possible improvement with regards to the possible improvement of gray matter quantification, gray matter atrophy quantification. As it is done for the brain, it might be rewarding also to fill the lesions within the cord to improve gray matters versus white matter segmentation, also in the structure and not only in the brain. So regional assessment also, there is a lot of work that is being done to not only to quantify damage within the spinal cord, but also to better localize this damage where it is likely to occur. Of course, it is difficult because the spinal cord is smaller, but there are some studies showing that using tractography it is possible to differentiate damage within the cervical cord, showing higher diffusivity abnormalities in the dorsal and lateral columns. Also, the atlas is developed by Julien and included in the spinal cord toolbox can be useful to quantify damage in the microstructural damage in the spinal cord in the different sections. We are applying them in a study, in a recent study that I am not showing here, but still work in progress in our lab. And with regards to possible post-processing improvements, of course, there are also some nice recent developments. For example, using deep learning, convolutional neural network that has been extensively applied to automatically segment lesions in the brain. There is also this recent application for the spinal cord that seems to be very useful and promising. And as I mentioned to you before, longitudinal assessment of cord atrophy is still a topic in which improvements can be done. For example, in the study, in these two studies by Ferran and Marcello, quantification of cord cross-sectional area was compared with a more sophisticated pipeline assessing the boundary shifts of the cord between baseline and follow-up, so suggesting that applying this method is more reliable than measuring cross-sectional area at separate time points when monitoring the development of cord atrophy, and all of these results in lower sample sizes for future clinical trials. So just a very brief look at other possible techniques. Spinal cord fMRI was already reviewed very nicely by Joe. We did some quite old studies 10 years ago using active task in the spinal cord, showing some abnormal activation, increased activation within the cervical cord in patients after a stimulation, tactile stimulation of the palm of the right hand. But in this field, newer studies are coming, assessing resting state functional connectivity, And the first studies are showing actually that there are some abnormalities of resting state functional connectivity correlated with the presence of lesions in the spinal cord of multiple sclerosis. And of course, other microstructural techniques, other non-conventional techniques assessing the microstructural damage can be used to, for example, myelin motor fractions, sudden imaging, spectroscopy. Of course, there are a lot of possible other non-conventional techniques that can be applied to MS to better describe damage. So to conclude, high-quality MR imaging is feasible in the spinal cord, especially in the cervical cord segment, has been applied to describe cervical cord pathology, and is improving our understanding of all the pathophysiological processes that are occurring in MS. And of course, there are still a lot of research topics that can be assessed with possible future improvements. So thank you.
So thanks for the very nice talk. I think we have time for one or two burning questions. Nothing? Okay. So maybe it's good if we move to the next speaker to stay on time with schedule, and if anyone has any questions, I think Paolo is there now. So thanks again. From the UCL and Siena University, and during the past year she has been very active in recruiting MOG patients. So today she will show us some results of her studies, and especially on the spinal cord imaging in this relatively new spinal cord disease. So thanks for that.
Okay, so thanks, Faran, for giving me the opportunity today to talk about spinal cord involvement in MOG antibody disease, and thanks, Marcello, for the introduction. So I'm a neurologist, but I have started in 2017 a PhD at UCI, and I'm going to show you some of the results of my research today. So we'll start talking about spinal cord involvement in MOGAD, then show some preliminary results, and I will conclude and give you some suggestions for future research studies. Some of you may not be familiar with MOG antibody disease. It's a new antibody-mediated disease, inflammatory-mediated disease of the central nervous system, characterized by the presence of a serological antibody against MOG, a CNS-specific protein located in the outer layers of the myelin sheath. So as you can see from this picture, the pathogenesis of MOG antibody disease is different from other diseases, inflammatory diseases such as MS and aquaporin 4 antibody. However, the phenotypes may overlap, and in clinical practice, the differentiation between MOGAD, MS, and aquaporin 4 and MOSD is not always easy. In particular, the spinal cord involvement with transverse myelitis is a hallmark of the three diseases. It is crucial to differentiate between the three diseases because prognosis and treatment are different. So the group in Oxford with Romina, who is here today, has tried to identify the demographic and clinical findings to differentiate between the three diseases, looking at the spinal cord involvement at disease onset in MOGAD. Transverse myelitis is quite frequent, in particular when the age at onset is in the adult phases. And transverse myelitis can occur isolated or concomitant with optic neuritis. Also, MRI over the last few years has shown some similarities and differences in the spinal cord involvement in the three diseases. In particular, in MOGAD, lesions can be short or long, but they predict the caudal part of the spinal cord from the thoracic lumbar cord and conus medullaris, and they are mostly located in the gray matter, forming an H dot. In NMOSD, the hallmark is the longitudinally extensive transverse myelitis with these lesions extending for more than three segments, and they are usually central. In MS, we know that lesions are more often short and predict the cervical cord, and they can be multiple and asymmetrical. However, always in this case, in clinical practice, it's not so easy to distinguish the three diseases. In fact, for example, I have just said that longitudinal extensed transverse myelitis is a normal of NMOSD, but in NMOSD, it has been reported that there can be short myelitis in up to 40% at disease onset. Also, the length of spinal cord lesions in NMOSD depends on timing of MRI. In this interesting case report, this patient presented with vomiting and hemiplegia, and at disease onset, it was visible on the MRI, only this lesion on the medulla. However, the lesion extended over time, involving the upper cervical cord area. But the upper cervical cord area extensive transverse myelitis was visible only 12 days after the disease onset. By contrast, in MOGAD, lesions are not always clearly visible and may disappear or resolve partially, giving this kind of picture like this dirty spinal cord sign without any lesion clearly defined in the cervical cord. So we can say that conventional MRI has only a limited capacity to distinguish between the diseases. Well, we have seen in these 2 days that cord atrophy has been largely used in MS as an outcome measure, and this is just one of the latest techniques that have been published recently by UCL. In MOGAD to date, there is only this study applying non-conventional MRI to the spinal cord. In this study, using the cross-sectional area, the authors found that MOGAD patients did not have cervical cord atrophy. Therefore, we decided to explore the role of new spinal cord imaging biomarkers in MOGAD when compared to aquaporin 4 and MOSD and relapsing remitting MS. To address this aim, we conducted 2 exploratory studies, one looking at cervical cord atrophy, and then we looked deeper in the cervical cord, applying diffusion tensor imaging and looking at the microstructure. This was part of a larger study which involved 2 centers, the Queen Square and the NMO Clinical Center in Liverpool. We selected from these 2 centers patients with the diagnosis of MOGAD, aquaporin 4 and MOSD, and relapsing remitting MS, according to the respective diagnostic criteria. All patients were outside the acute phase for at least the previous 6 months, and adults at the time of the MRI. We measured the cervical cord area with the GIM, with the semi-automatic method already described yesterday, and we also measured the cord volume automatically using spinal cord toolbox. For the DTI analysis with the help of Francesco Grustu, we processed the images using Aspartel and spinal cord toolbox, and diffusion metrics were calculated at the level of C2, C3. So this is our study population. We found 30 MOGAD, 30 NMOSD, 31 MS, and 34 healthy controls. There is a mismatch in the sex, age, in the demographic characteristic of these patients as for the respective characteristic of the diseases, because they have different disease onset and sex predilection. As expected, MOGAD patients were the youngest patients with less disease duration, lower disability. And at the time of the observation, we found only one patient with core lesion at C2, C3, compared to NMOSD and MS, where the number of lesions at C2, C3 was higher. So when we looked retrospectively at the disease course and the characteristics of our patients, in the MOGAD group, most patients had a relapsing disease course. Regarding the disease onset, as previously described, the characteristics of disease onset of our patients were mostly an NMOSD spectrum, since all of them were adults. There was an abnormal spinal cord MRI at disease onset in 6 patients with MOGAD, and an history of transverse myelitis during disease course in 63% of patients. Well, we looked, as I said, firstly at the atrophy, at the cervical cord. In all the 3 disease groups, we found lower mean values compared to healthy controls. But when we compared the disease groups and the disease groups with healthy controls, we found, as previously described in the only atrophy study, that in MOGAD, there were no difference between MOGAD patients and the other groups. The results of the volume mimicked the results of the area. When we looked at the DTI at C2, C3, we found that although the cross-sectional area did not show any difference between disease groups, we found that there were differences in the fractional anisotropy, in all the DTI measures. In particular, there was a reduced fractional anisotropy and increased mean diffusivity in the NMOSD compared to MOGAD, although there were no differences between MOGAD and healthy controls. So to summarize the results that I have shown today, we can say that our study confirms that the mechanism of spinal coordination in the 3 different limiting diseases is different. In aquaporin 4 and MOSD, the cord seems to be more damaged than MOGAD and MS. However, in MOGAD, and also in MOGAD, the spinal cord seems to be preserved outside regions affected by lesions. However, in MOGAD, we need further studies with larger sample size, and I think that we need to consider 2 main factors in the future study. First of all, the time of MRI acquisition, because we saw that the behavior of lesions is different in MOGAD and NMOSD, so the lesions can change, can evolve. And also, we need to consider that the location predilection, the lesion location predilection in MOGAD is different from NMOSD and MS. To support these 2 suggestions, I would like to conclude showing you a case report of a patient that we saw at Queen Square. This is a 32-year-old patient who came to our observation with a very bad transverse myelitis with severe urinary, bowel, and sexual dysfunctions, diplopia, and vomiting. The MRI at disease onset showed this diffuse, confluent, and bilateral hyperintensity and multiple lesions in the thoracic spine and in the conus medullaris. He was tested for MOG and found positive. Then he started the treatment with steroids and immunosuppressant drugs. He had several follow-ups over time in our clinic, And when, after 1 year, we repeated the scan, the lesions resolved over 1 year. However, the patient kept saying that he had sexual and urinary problems. So what was happening at the cord, although we couldn't see any particular abnormality? Then we decided with Marius to try to apply the study that yesterday Dr. Paniker showed largely. So we looked at the lumbosacral enlargement, and we compared the values of our patient with those described in the healthy control group. And in our patient, although MRI appears normal, the area and the MTR values were all reduced in both white matter and gray matter. So this suggests that beyond what we see on conventional MRI, there is something that goes on. And we need to develop MRI biomarkers that can be as specific as possible for MOG antibody disease. So I would like to conclude. Thank you, everybody, for the attention. Thanks to the MMR unit at 7th Square, in particular for the spinal cord study, Francesco Brusco and Isabel Della Paz, and my supervisor and mentor, Professor Ciccarelli.
Thank you, Hannah, for the talk. I think we have maybe time again for one question, then there is the coffee break to have a talk. Yeah, there is one question from Umberto.
Thank you for the interesting presentation and clinical, which is important presentation. Do you think we will need to do a more antibodies in the clinic, like, or we will find a way to use MRI instead of because basically now we always do a proper report and we mock, but not so frequently, and they're quite expensive for the sake. So we will find a way to really use MRI to do.
Yeah, I mean, the aim is that one, to find like some MRI findings that added to clinical features can lead the clinician to select patients to test for CDA. Also because the test, the immunological test is not available worldwide, and the sensitivity and specificity of the test is variable. So yeah, for sure we need to find some MRI biomarkers. Thanks.
Okay, time for the coffee break.
So this session will be chaired by myself, Francisco Bruce Rosa Cortese, and the first speaker is Olivier Vincent from Polytechnique Montreal, and he will be speaking about automatic segmentation of spinal MS lesions, how to generalize across MRI contours.
Thanks for the introduction. So yes, I'm a student with Julien, and so as you've probably noticed, nowadays almost every field has been invaded by deep learning, and MRI is not an exception of this. So we've discussed yesterday a bit spinal cord segmentation. In my case, I'm interested in MS lesions and using deep learning. So people sometimes are scared by deep learning because it looks like some black box that we're not sure what's going on. So I'll try to explain to you what we did and how it works and why it's interesting. So as you can see here, the number of ISMRI abstracts has exploded regarding deep learning in the last few years. So I'm surprised to be the only one at this conference talking about this. All right, so first of all, most studies regarding MS lesion segmentation have been done in the brain. So this one here is from a group at McGill. And essentially what they've studied is where does most error and where does the uncertainty occur. And what they found is that it's mostly for small lesions and at the boundary of bigger lesions. So what they've shown here in this figure is that here in A and B, these are really small lesions. And if you vary really slightly some of the parameters or threshold of the model, so the baseline is 1, and if you vary really slightly, this lesion here in A will simply disappear, and it's the opposite in B, it will appear. So this can be a problem because, well, at first, some might say, well, it's a really small lesion, who cares? But in the spinal cord, this is really important, and it can have a big impact on the prognosis. So this is some work from my colleague at the lab, Charlie, and essentially what he did, I think Paula talked a little bit about it. It's segmentation in the spinal cord, and the biggest challenge is the class imbalance. So essentially what this means is that the proportion of the image that's interesting to you is really small. It's like less than 0.1%. And this is a problem with deep learning because you're looking for something that's like really hard to find. So what he did was a model that first it detects the cord, and then it does 2 steps. The first step is to crop the image around the region that interests you. So this reduces the class imbalance, and then you can segment it using a normal CNN model. So he got pretty good results, and overall, it's the same conclusion as in the brain. So the biggest difficulty between the manual and automatic segmentation is at the boundary of lesions. So you can see here between the manual and auto in both sagittal and axial. So yeah. So where does this difficulty come from? It comes from the fact that deep learning has problems with generalizing to images that it has never seen before. So here, this is just to show that between subjects, if you keep the acquisition parameter fixed, as Julien discussed yesterday, there's not much variability, so that's not a problem. The problem is when acquisition parameters are varying and create lots of different contrasts. And if it's something that the model has never seen before, it's not going to be able to generalize well. And this is problematic, especially in multi-center study, because you're going to come across something you've never seen before. And yeah, so all the imaging parameters that can vary a lot and create all these different contrasts. And that's one of the biggest problems as to why deep learning is hard to apply in real life. So the idea of our project was to, instead of simply, usually what you do is you take the image, you give it to the model, and you ask the model to give you a segmentation. Well, instead of doing that, what if we also added the parameters? You give more knowledge to the model about the MRI physics. And this can be really hard because there are many parameters and like an almost infinite number of combinations, and it's impossible to have a data set that covers everything. So a more tractable way to do this is to start by giving to the model what's the contrast type of your image. So you give to the model an image and you say, well, this is a T1 image. And that's what we did. And that's usually what a human will do, like without even realizing. It'll look at the image and say, well, this is this type of image, is this type of contrast, so now I can segment it. And so the way we did it is that you have the classical model, which is a UNet. It's the gold standard in segmentation. So it takes the image and it outputs segmentation. And this is what's called the film layer. What it does is essentially it modulates the segmentation of the UNet. So it takes the contrast type and it learns what feature of the image is most important based on the contrast type. So it'll say, well, since it's a T2, this feature is more important, therefore you higher important. Well, yeah. So that's the idea. We've used data from 13 centers with over 30,000 slices. As you probably know, the deep learning requires a huge amount of data to work. And it's a large range of acquisition parameters. It's a mix of T2 and T2 star images, which were all manually segmented for MS lesions. So first of all, we wanted to test, well, is there really a generalization problem? And the answer is yes. So as you can see here, if you train your base model, so just the segmentation part without the contrast information, if you train it on T2, you'll get roughly 0.55, and on T2 star 0.59. So you would have expected that when you combine both, you would get something that is in between because you've trained the model on both type of images on the entire data set. But that's not what you get. Essentially what we see is that when you train the model on both type of images, it fails to learn the things that are the additional information that's in the T2 star images. And that essentially confirms the difficulty to generalize. And now if we add the film layer, we get roughly 5% boost in dice score, which is pretty good. And it shows that simply by saying to the model, this is a T2 and this is a T2 star, it can learn to better differentiate by itself the feature in each of these images. So this is an example. Here it switches between the ground truth and the segmentation. So as you can see, this small lesion here, they don't agree on the positioning of the lesion. So this is consistent with what we've seen previously on the brain. However, another thing we found is that after optimizing both models, like pushing the limit, both models reach a ceiling at around 72 of dice. And this is surprising because at first we had like a pretty good difference between both models of 5%. And it suggests that maybe there's like a global limitation inherent to the data set that's independent of the model we use. And we think inter-rater variability could be the answer to this. So inter-rater variability was estimated to 0.61 in our data set, and we were already at 0.60 using film. So what's probably going on here is that we're overfitting and overlearning a certain rater that has possibly rated like the majority of the data set. And I think it's interesting because going back to the study in the brain at the beginning, which showed that most uncertainty occurred at the boundary of lesions and in small lesions, intuitively, this is where a human rater would hesitate the most. So it's possible that, in fact, when we think that the deep learning model is committing mistakes, it's not actually a mistake, and it could be a valid segmentation for somebody else. So what would be interesting is eventually to add knowledge of the rater into the deep learning model. And the way to do this could be with film. Essentially what we did is we said to the model, well, here's an image, it's T2 segmented. Well, we could do here's an image, it's a T2, and it was segmented by Bob. So can you segment it that way? And eventually the model could learn the different rater style. And this could be used to show that indeed the limitation doesn't come from the deep learning model, but from the inter-rater variability. So in summary, what we showed was that there is indeed a generalization problem, and it can be solved in part by adding knowledge of the contrast type, such as with film. But we also showed that the real limitation, once you optimize the model, is not really the architecture, and it's mostly found in the data set. And that's interesting because you'll see like lots of contests and challenges who are trying to push the limit to the maximum of like how high can it go. But at some point you have to ask the question of, well, is this additional performance meaningful? Because once you go past the inter-rater variability, it's probably not super meaningful. And so that's what I found interesting in this. And yeah, in future step, I think it would be really interesting to add this inter-rater uncertainty, because you could eventually say, well, if you manage to learn the rater style, you could say, well, I'd like to segment like Joe because Joe has like 50 years of experience. And so this would be really interesting. So I'd like to thank everybody in the lab who helped me, all the collaborators for sharing data. And yeah, Julien for bringing me here today. Any questions?
Very nice talk. I think that if you probably got two raters doing the same and calculated their size, it would be the same as your performance of your deep learning. So, but one thing that maybe we haven't thought of yet is to ask the rater to improve their own uncertainty. So rather than, so if you've got a rater that at some point we'll have to make a decision, but when you draw lesions, I'm sure that at some point you think, okay, should I go there or should I include or not? And then you make a decision. But you have a very, I mean, I think the raters will have a very self-confidence interval that they are following somehow. So maybe you could say that if you could have some raters that say, okay, this is 100%, this is the lesion, but in this case, this is the lesion, but the lesion could be this. And then I don't know how you can include that information in your deep learning. That's your problem, not mine. But ask the human person that is rating. Because at the moment, you go with the one drawing that you assume that for that kind of person, for that rater is absolute. It's definitely not absolute.
Yeah, or have 10 or 100 people do it, and you create a lesion probability map, and then you have an uncertainty estimate.
Yeah, depends on what is easiest. But then there may be some areas that everybody rates as uncertain. So it would be interesting also, which could be a class of your output.
Yeah, really interesting point.
I don't know. But it's something that I keep thinking.
First, we'll go on the back.
Yeah.
Nice work, thanks. Can you tell me which images were used by the raters to subject?
Well, you don't always have both. Maybe Julie can chime in because he's more familiar with the data set.
So we had T2 and T2 star. They were both segmented by raters, and then they were merged, like co-registered, merged together, and the lesion were kind of averaged. Like the lesion masks were kind of averaged across contrast, and that was used as a ground truth. And there was, and to follow up on Claudia's very good comment, we also had two classes, like a very like 100% certainty and half uncertainty about lesion masks. That was not used in this study, but we have the information.
No, it's just because I was surprised that P2 star gave better performance than the P2 one of the first results slides.
Yeah.
Almost as good as the final result where you have this film.
Yeah.
Yeah, but I think that is what you also see in clinical practice, because on the T2 star you have more homogeneous CSF signals, so then you have a nice homogeneous. So many visual studies also find equal or not even better performance for T2 star compared to T2 reporting.
And if I could follow up, the final result that you had was even much higher. The dispersion went from 0.55 to 0.6, and then suddenly you're at 0.71. So what did you do?
Well, it's essentially just running, like, you have lots of parameter in deep learning, and you just like do a grid search and just wait until it finds the best parameter available. Yeah, pretty much.
Images were acquired in the same direction. I mean, all were axial, or you have, for example, the T2.
We're using axial in
axial.
Yeah, axial in this.
Both. The T2 and the T2 star.
I think so. Yeah.
Yes. So we had also sagittal T2 weighted images. I'll have to check for this particular study if we like removed them or not, or if we included them. That was like my answer to Hugo, is that we had sometimes both the let's say the axial T2 star and the sagittal T2, and we did the segmentation on each of them and then merged the segmentation. Yeah, we'll have to check whether this was axial only or not.
Because I imagine that this has an impact as well, because when you draw the lesion, it's not the same. Well, you will see the same area in both images.
Yeah.
Yeah, so you looked at the dice. Did you also look at the number of lesions? How correct you were in finding the number of lesions rather than the exact outline of the.
It's something we're looking at currently, adding more metrics like lesion-wise and volume-wise. So it's something we're currently doing. But I don't have. I don't have the numbers.
Without lesions, for example.
Yeah.
Binary classifications.
Could you just step through the architecture in a bit more detail? So the inputs, you've got a traditional UNet taking T2 and T2 star as the first stage.
Yeah.
And then the film stage takes what, T2, T2 star again, plus the segmentation, plus the contrast types?
The film only takes like a one hot encoded vector, and it's just like, is it T2 or is it T2 star?
Right, so it's got the, but is it also seeing the input image and the segmentation? Or?
No, only the type here. But both will learn at the same time when you do the back propagation.
Right.
So you've got like both neural network here, and they'll learn at the same time.
Okay. I need to read your paper.
One question there. So you're trying like to generalize in the way that in the same neural net, inserting T2 and T2 star images.
Yeah.
And the idea is to have the neural net work properly for both.
Yeah.
Processes, no?
Instead of having like two separate networks. Yeah.
And why? And I mean, is there an improvement with respect to two separate networks? Like, yeah, because.
Like these are the two separate networks. So there is an improvement because you're learning from more data. Like you're learning from twice as much data, and so you can get better knowledge of.
Did you like it would be interesting if you can take a look deeper into seeing if the layers weights and how they differ between these two nets and the general one, because maybe I don't know, maybe there's like there's probably they're learning the same features until a certain point, and then there's like some differences,
And I think it's okay, it can be a good approach to focusing on the like, okay, these are like the three networks are learning in the same way for these features, but for these features, they're learning independently different. So maybe that can be like, oh, maybe I should focus on like the differences.
Yep.
Like on the scene, like on the layers.
Yep, for sure.
Right.
Maybe just like a very quick follow-up on the comment of, why not providing two models, 142 and 142 star. I mean, this is like a dummy proof of concept, because down the line we have like thousands of different sites around the world, each using different parameters, and we don't want to ship 2,000 models with SCT, because it's just going to be chaotic. So in the ideal world, we have one model that works for all.
Yep.
I think we can move to the next speaker now. Let's thank the speaker.
Thank you very much for the invitation to allow me to give an introduction of the study that we've done years ago. And in retrospect, it's kind of complementary to the reproducibility data that Julian introduced yesterday, and hopefully also addresses the question of the repeated scans on the same subject question raised yesterday. So just to review that, this study was supported by an infrastructure grant by the IPMSA, the International Progressive MS Alliance. So it's fairly focused on progressive MS. And in the field, if you are a clinician, you know those progressive MS patients are not common to find, and that comes the need for multiple sites to participate in the study. And we need quantitative biomarkers other than brain MRI, because those spinal cord scans can directly relate to the functions in the sensory and motor system, and it could advise you to make a decision avoiding the complications of the crossing fibers and other issues in the brain. However, we are not there yet, because the reliability of the spinal cord imaging, especially diffusion, is not there yet, and we are catching up with the brain measures in both the atrophy and the other advanced imaging biomarkers. And you will see in later part of the study that we have a particular bias towards diffusion, because that's what we are mostly interested in. So the study design is a little bit complicated. And it's on a small scale, but hopefully we can still get some insights from these limited data. So we recruited in total 6 healthy controls and 6 primary progressive MS patients, and the 3 subjects in each group from each site. And the healthy subjects traveled to the other site at a short-term interval. And also on 6 months, they traveled again to the other site. The patients never traveled in a long term. The patients also traveled to the other site at short term within 2 months, but they never traveled at a long term 6 months because we don't know what may have progressed during that half-year time scale. And each subject, they are scanned within their own site within 2 months. So we have intra-site reproducibility. So just to, so that gives us about 37 total data sets that we are happy to release and soon to be in open neuro. they are packaged in GIT data format, and hopefully you are able to simply easily digest intake those data from OpenNeuro. And these are the conditions at Mount Sinai and at Washington University in St. Louis. Dr. Inglesi and Dr. Naismith helped to recruit the healthy controls and the MS patients. Oops, sorry. To digest a little bit about the study design, so you can see at maximum, a single subject could have 6 scans, 2 scans in the same site here within 2 months, and 2 scans on the other side in 2 months. And after 6 months, they scan again in the site, and they also travel to the other site for another scan. However, not all the subjects travel from one site to the other site, so it's asymmetric. Only site one travel to the other site. We think that it's enough because budget limitation and the logistics, and we hope this is good enough to give us reproducibility data with repeated scans. So from these data, we can get some intra-site short-term reproducibility by analyzing the data in this group. And we can also get intra-site short-term within 2 months by grouping these data. And we can also get intra-site long-term separated by 6 months. So these are the reproducibility metrics we are analyzing in the later part of this the talk. In terms of the protocol, we have a T2, fairly standard, a PSIR, a single slice, was originally proposed by the UCSD group, 3D MT transfer protocol, and we spent a lot of time on diffusion. I think just by my personal bias and the group's particular interest in diffusion. We have 5 millimeter covering C2 to C5 continuously. We scanned both base encoding AP and the PA with 3 averages. So that itself is about half an hour scan diffusion. We intentionally oversampled to have enough data later if we want to combine data, come up with new ways of distortion correction. We can always play with this data. And in addition, we also have thick slice 10 millimeter individually angulated to be perpendicular to the cord, and that has a landmark in the center of the vertebral body from C2, C3, C4, and C5. These scans each take 1.5 minutes, and we also scanned reverse the base encoding direction with 2 averages. So if you want to look at the effect of SNR for some advanced diffusion microstructural modeling, you can use this data and compare with the 5 millimeter data if you want. Oh, by the way, the 2 scanners at Mount Sinai is Siemens Skyra, and at the time at WashU it's Siemens Trio. First, the structures, nothing particular fancy. We just did the core segmentation with spinal cord toolbox. At that time, we don't have the automated segmentation of gray and white, so we just did gray and white. The results, you can see there's no phosphedrine use. It's pretty consistent with what has been shown yesterday and today. And if you want to look at the difference between the 2 groups, patients and controls, and you can see indeed there's difference at group level, but there's exceptions. Some of the primary progressive subjects, they don't necessarily have a very particular atrophic cord. Now comes to the diffusion. Our diffusion protocol is a bit different from the standard protocol that Julian has putting the common consensus. So I'm going to take a bit of time to introduce it. It's an old school inner volume imaging diffusion sequence, and we put the second diffusion, second inversion pulse at the beginning just to reduce the time between the two 180-degree pulses to reduce the lost magnetization. And we introduced a bit of a multi-band factor to alleviate the problem with the inefficiency of the inner volume imaging. The resolution is the same as the common protocol, 0.9 in plane with 5 millimeter thickness, C2 to C5 for 12 slices, and we did a quite full harmonization. First of all, the sequence itself, it's source code level harmonized. The source code has been integrated into the CMRR multi-band public release master code base, so we can have the same sequence with the same source code for various different software version Siemens platforms. And we particularly matched the echo spacing, almost all the details, even the small delta and large delta of the diffusion encoding. We use linearly spaced multiple p-values, and each p-value have unique directions. And the acquisition is slice by slice cardiac aided. So all these data is going to be available, and you can analyze these multi-p-value, multi-direction diffusion with your favorite modeling. And for the rest of the talk, we only take a part of the data with a single phase encoding direction, and only 5 millimeter data. This is close to what, in a realistic situation, that you have 10 to 15 minutes devoted to diffusion. And just to show you the quality of this slightly accelerated diffusion protocol, even with a non-optimized coil, because there's no Z separation for the neck coil, for these coils, at least the neck on the Spectral and on the trio. And you can see that the slide shows no artifacts, even with this very short Z coverage, which is not ideal for multi-band. And we've shown this very minimal residual aliasing. So these are the two simultaneously applied slices, and the eye pulse for the other slices turned off, so we can assess the leakage of the signal from one slice to the other slice. So this is close to the noise level. And if you turn up this a little bit more to multi-band 3, you can see the artifacts coming out. So we are confident that the acquisition, even with slight acceleration, can give you nice results. I'd like to take the opportunity. Dr. Jung-Won Kim used to be at Sinai, and now as a junior faculty at Baylor. He has been instrumental for all the diffusion MRI, QA, and the pre-processing and the analysis in the rest of the slides. And we take quite a bit of time, go through all the slices, actually each image, going and identify these artifacts in the diffusion. And these will be encoded in a specific file in the data release to identify these. If you want to do your modeling, it's recommended you take these slides out at that particular diffusion direction. So we can generate DTI parameters here. And working with Dr. Vic Song at Washington University, we're also particularly interested in multi-compartment modeling with these multiple p-value, multiple direction
data sets. And the particular model we're interested in is called the diffusion basis spectrum imaging. It is a multi-compartment model that takes in a spectrum of diffusivity with a single tensor. So you can generate in the tensor modeling, you can get similar to the FA, RD and AD. In the DTI, you can get these parameters specific to the fiber compartment, which is illustrated here. In addition, you can get these isotropic components, which is also what the DBSI models. So these are compartmentalized into water fraction with close to free water diffusivity, restricted fraction with a little lower diffusivity group, and the hindered fraction, which is a really low diffusivity. So that's where the name comes from, because it has a spectrum of these isotropic components. And we also did, before we ditched our years ago protocol of manually defining ROIs, we also did the automatic ROI. This is something probably for later discussion, that we did find a little bit slight bias by these automated ROIs, and it's in the expected direction and confirmed by the DBSI fiber component. It's these voxels at the edge of the cord, and also particular at the junction of the dorsal horn. There could be potential improvements of the automated ROIs that we need to clean up. The effect is not large. If you look at the scatter plot of FA with automated versus manual ROI, but it is there. There's a defined bias. Although it's not our primary interest to compare healthy control and the PPMS, but it's a good sanity check to see our values go in the expected direction. Because we don't have a lot of subjects, so the p-value doesn't make very precise statistical sense, but you can see from these, just plotting all the data, you can get a sense of the trend that the patients has a higher diffusivity and lower FA. If we do estimation statistics, so you can see bootstrap the confidence interval for the difference between groups. So this is good. We are doing the quantitative number we get, either DBSI or DTI, is making sense. The more interesting question is, what are the variances? Because that's what the purpose of this study. We want to extract the variance and extract the variance and partition the variance so we can inform a longitudinal study. We used the R and a linear mixed effects model package LME4. We confirmed the fixed effect, as other people have also reported, and also we've observed that the posterior column and the corticospinal tract, they are different. The difference is small, but it's definitely statistically very significant. Especially if you use linear effects, linear mixed effects analysis without just averaging all the data together. And there's the expected side difference. For the random effects, we noticed something interesting, that we put the slice nested in the subjects, and we noticed the slice level variance is at the same level at the subject level variance. This is, if you're not particularly familiar with the variance, I can say this is a much higher level slice level variance compared to what you would get from brain diffusion MRI. So that just informs you that getting more slices in the spinal cord diffusion MRI is helpful for you in the statistical sense. And this is the the need of the analysis, the variances across time points. First of all, we didn't find a particular difference between these time points, the baseline 2 months and 6 months. So for our later summary plots, it's okay to average across time points, but more importantly, we can estimate this variance by leaving out the variance across here at the subject level. So this is the random effects that will inform us for power analysis for longitudinal study. And this is the kind of power analysis that we hope to get for sample size estimation for larger studies. And for example, if you expect to detect a 1.7% per year progression of the FA reduction by using some of the others measure in the brain, in the corpus callosum from this study, you can see if you sample follow subjects and collect data every year, you will need about 200 subjects to use the spinal cord diffusion MRI as your biomarker for the sample size. And if you measure every 3 years, you can reduce it to 40 subjects. And this is the information I think will be very helpful for neurologists. And this is what we are aiming for in future studies. Look a little deeper into the reproducibility as measured by coefficient of variance. These are the box plots across subjects. And you can see the intrasite short-term and long-term, they are quite similar. But because of the site bias, the bias was not taken out here. So it drives up the coefficient of variance between sites. But this is potentially correctable if we can estimate the fixed effects between sites. So we can potentially take it out and recalculate the coefficient of variance after the correction. Hopefully this will drop down to similar level as intra-site. However, we haven't done this analysis yet. And one particular surprising thing is we noticed that the radial diffusivity is particularly more irreproducible than the other measures. It's a little bit disappointing because in our prior cross-sectional studies, we find the radial diffusivity is the best correlates with quantitative sensory measures such as vibration sensation or the time to walk. So this is maybe some consideration for the design, how useful the radial diffusivity is in the longitudinal study versus cross-sectional study. In terms of the DBSI measures, it's the same similar pattern that the intrasite is pretty good, and site-wise bias drives up the coefficient of variance. The same thing happens for the radial diffusivity in the fiber compartment of the DBSI. But very gladly, we found that the DBSI reproducibility is similar to or even better than the DTI, which is at the beginning a little bit surprising. We thought that it's a more complicated model. You have more unknowns. It should be less reproducible. We're still digesting why it is so. It may be that the mathematics between within the modeling that passes out the more irreproducible variance into the components that we are not shown here, which is the restricted compartments or free water compartments. That may explain these results. In any case, so all the data will be available. And personally, I like to drive this into a more, even more precise and more reproducible in the next 5 years by further harmonizing higher performance and 3D system that's not available 5 years ago. For example, we can achieve the same, even better data quality as the Prisma scanner. and push the resolution even further to 0.75 millimeter. These systems have a stronger and more stable, stronger and stable gradients, and it's likely to get long-term support by the vendors. So these are good for progressive MS studies. And the phase 1 probably organized 3 or 4 Prisma sites and expanded a dozen sites with high-performance 3D system. And you know that diffusion MRI of the spinal cord is lagging behind a lot from the atrophy measurements, not only to the brain, but also atrophy in the spinal cord. So I want to see that in the next few years, we can push this to be highly reproducible to rival the atrophy measurements. So the diffusion MRI can contribute its due value to the MS research. And hopefully we can get about 10 progressive MS patients per site scanned yearly, and we will, by that time, end of phase 2, we will have about 100 progressive MS patients readily available at any time points, so we can, people can design very flexible clinical trials. And I'd like to thank the IPMSA that supported this very preliminary pilot study. And thank you for your attention.
Questions from the audience? Yes.
Yeah, thanks. Very impressive work. Very nice. I like the work also where you showed the variation. Slightly surprised that it seemed to be that the long-term disability was better than the short term, and go to the, at least the previous slide, I think, behind the left two plots, where the sort of the size of the boxes, and that you understand why that would be, or it seems a bit.
I mean, the short term is larger than the long term. I think it's because we don't have enough data. It's just inherently we don't have enough data sets to make these box plots that's reproducible enough. It has its own variance. If you look at the specific data points behind these box plots, it's not a lot. Okay. Yeah, it's less than 10 data points. Yeah, because we only have total 12 subjects, and not all of them travel to the other sites. So it's so if you look at the p-value, if you really want to compare the p-value between the COB between these two, I believe the p-value is not going to cover much.
And if you compare the automated and manual ROIs, that's what this refers to, right?
Right. They're comparable.
Yeah.
Reproducibility is comparable.
It seems to be that reproducibility is better for the manual. Is that because the automated is more variable, or because, like you said, it sometimes takes?
I wouldn't say the manual is more reproducible. They are probably on the same level. What worries me is the slight bias. That
Can you actually go back to how you selected the manual versus the automated one, and on which? Are you doing this on the V0, or are you doing this on?
Yeah, we are guided by both the FA and also over data with V0. We want to identify the central horn, and most of the time we can. And at that time we have we draw these 40-degree lines to the suspected location of the dorsal horns, and we intentionally avoid the voxels next to these dorsal horn lines.
That image looks much higher resolution than the automated.
Yeah, it's oversampled, twice oversampled in fact. Maybe I'm not sure.
Is it post-processing, or is it, did you acquire it or something?
No, it's we oversampled in the diffusion data space. Not oversample in the FA, because that will blur out these edges. In the diffusion, in the DTI raw data space, it's smoother. So we oversample at this stage and calculate the DTI.
later.
Yeah, this is oversample,
Yeah.
so it has twice the in-frame resolution than acquisition.
But if you feed that image to the automated drawing, wouldn't that do better as well? Or, you know, why did you not use the same background image for both? You know what I mean?
Yes, I understand what you mean.
Seem the same. Okay. And also the other comment was that if you use FA to draw your regions, and then you are going to measure differences or COVs of FA, of course, if you have highlighted by manually selecting the high FA voxels, you introduce a bias.
Yeah, we understand that. Yeah.
You increase your reproducibility just by it. So I would say that could introduce biases in measurements, especially in patients. So.
Yeah, indeed. Yeah, we want to move away from the manual to here. It's just this observation we noticed. In the, I think this is the default without oversampling, right?
Yeah, we need to look at the details.
Yeah, so we just note. I'm not suggesting the manual is better. We indeed want to move it here, but in the progress, in the process of doing so, we notice this observation. So there's some improvements we can make by combining the experience that we had at manual here and try to avoid this bias.
Yeah, just a quick question on the. So it's more on like a comment on the reproducibility. So I understand the DBSI seems to be like more precise. I just wanted to make a comment on the ultimately precision is one thing, but the normalized effect size is another one, right? And so ultimately you could have like a flat metric, right, that is highly reproducible, but if it's poorly sensitive to pathology, then that's also irrelevant. So my comment is about, you know, how do you see the DBSI being more reproducible, but potentially also less sensitive to pathology, and in relation to that, what's your strategy to choose the p-value which also impacts the reproducibility, but at the same time the sensitivity to pathologies?
To answer your first question, this plot informs a little bit about the sensitivity of the DBSI, which is here. So it shows the same pattern of this shifted value in the patient group versus control. So that lessens your first concern. The second question about the influence of the p-value and the diffusion code, the details of the diffusion encoding, I think it needs densely sampled, oversampled data sets to do a fair comparison. Here, it's inherently not apple to apple because the same amount of data used for DTI modeling versus DBSI modeling, they are just in itself not comparable in a modeling sense.
Regarding the p-value, I have a comment. I think you show that it's b maximum 1,000,
right?
And so at b 1,000, you're not really sensitive to much to non-Gaussian diffusion, but DBSI is really looking for, you know, multiple diffusion components, so really non-Gaussian diffusion. So it's a bit surprising that you can simply fit DBSI to such data set. Doesn't really look like designed for non-Gaussian.
You have some misunderstanding of the DBSI, which is different from the others and other diffusion modeling that model the non-Gaussian portion. Everything here is modeled as Gaussian.
Yeah, but some of Gaussian diffusion components results in a non-Gaussian overall behavior.
You mean at the 1,000, some will have non-Gaussian.
It's very difficult to separate non-Gaussian diffusion at B1000.
There may be some, but I don't think at 1,000 you have particular sensitivity to non-Gaussian. But I think it may be absorbed into the hinder diffraction, which is very small dots here. That's modeled as cells here. So maybe some of these components will absorb non-Gaussian signal properties.
I think we need to move on. Stand, let's thank the speakers again.
Let's move on to the next presentation. Robin Nall from National Institute of Neurological Disorder and Stroke from the US. We'll talk about assessing the spinal cord atrophy in neuroinflammatory disease.
Thank you for the introduction and thank you for inviting me to this workshop. I think this has been a great experience so far. I'm going to talk about assessing the role of spinal cord atrophy in neuroinflammatory diseases. And one of the diseases that I'm going to talk about is multiple sclerosis. We have talked about multiple sclerosis all day today, so I'm going to keep it brief. Just to say that it's an immune-mediated disease of the spinal of the CNS. Its characteristics is lesions. But also brain and spinal atrophy is implicated in this. One of the presentations is chronic progressive myelopathy. The other disease that I'm going to talk about is HTLV-associated myelopathy or tropical spastic paraparesis. It's a mouthful, so we abbreviate it as HAM-TSP. It is caused by a retroviral infection, mainly targeting the thoracic spinal cord, and it's non-specific. Brain lesions are characteristics, but also spinal cord atrophy. This also presents as a chronic progressive myelopathy. So the idea here is to learn from other diseases so that that has a direct implication in the thoracic spinal cord, and we can bring that information back to the MS world and see what additional information we can go from there. So just a few words on HAM-TSP, HTLV1 or human T-cell lipotrophic virus type 1. Was the first retrovirus identified in humans. It incidentally happened in NIH in the 1980s by John Callow's group. It's endemic in many areas around the world with about 15 to 20 million worldwide infections. It's endemic in Japan, Australia, and the Caribbean, but also large populations of South America and some countries in Africa also carry the disease. It is transmitted through bodily fluids, and luckily, 90% of those infected are asymptomatic. So, so asymptomatic carriers are denoted in this rest of the presentation as AC. The rest of them develop the HAM-TSP. It is also implicated in adult T-cell leukemia, in which it was first identified anyway, and other inflammatory manifestations include uveitis and myositis. Here's why we are interested. So NIH has one of the largest HTLV clinics in North America, but to this date, was some of our patients present with the clinical diagnosis of progressive MS. This is an example. This patient noticed paresthesia in her legs in 2004, developed weakness in her legs, trouble walking in the next 2 years, started to use a cane in 2007, walker in 2009, and a wheelchair in 2018. And when she came in, she had a diagnosis of primary progressive MS. We did the HTLV1 test. We got a test for HTLV1 virus, and we saw it was positive, so she had a completely different disease. So this is the important. So the virus integrates into the host DNA as a provirus, and that is how we can test for this. It predominantly infects T cells, so we can get immune cells from the blood or from the CSF to test. And you can see that the HAM-TSP has detectable virus in the immune cells. So this is as a percentage of cells infected. Clinically, as I said, the disease localizes to the thoracic cord. We have patients present with back pain, weakness, stiffness, all the things that we saw yesterday from Dr. Panikkar. and also urinary involved incontinence, sexual dysfunction, etc. And like any other neurological disease, it has varying progression. So in some patients, it progresses rather mildly, with little or no progression seen in symptoms over 20 years. But others, it progresses very rapidly. A patient going from being able to walk to a wheelchair ridden in less than 10 years, just like the patient I showed in the previous slide. So autopsy tissue, we can do pathology, and we can see that in this example, a 59-year-old male who had severe dysfunction has much thinner thoracic cord than the 75-year-old female with moderate dysfunction. So I think this is very evident. The atrophy is very evident, and also maybe you can see that there is some myelin pallor in the lateral columns.
Is there some selective sparing of the posterior columns?
Yeah, there is. It seems to be, which is yeah. So sensory is fine. So it is not totally spared. There are, we saw that paralysis, but motor is much more affected. And we can do some immunology. So this is the thoracic spine again, and you can see all these dots are CD8 positive cells, which are essentially antigen-specific cytotoxic T cells. So these cells are recruited to an area, and if they see cells presenting antigens, so they induce apoptosis in the cells. So there is tissue death in this. So this is direct cause of tissue death can be attributed to CD8 positive cells. And of course, CD4 T cells are also present in this area. Like I mentioned before, the MRI in the brain shows some non-specific white matter diseases, lesions, but overall, not too much going on in the brain. But if you look at the spinal cord, we have there's a healthy volunteer here, and this is MTSP, and you can see visually that maybe this is thinning. So that is what we wanted to quantify, of course, and we developed this. I think this is the sixth method of spinal cord cross-sectional area or atrophy that we have seen in this workshop, but nevertheless, we use a volumetric T1 weighted image using 3D GRE sequence, and we acquire the cervical and thoracic cord separately using the automatic table move function of the scanner. That means that we can use the DICOM to stitch back this cervical and thoracic cord here. And then we use MATLAB scanning function, which is again a gradient-based method to detect the edges, all the edges of this image. The only manual input that the user has to give is identify the edge that is directly behind the C1 and T10 vertical columns. And then the program fits the spine to this, reformats axial slices from every single point along the spinal cord. This is manually drawn, so it's not perfect, but this is done using the slope of this function, so it is very accurate. Then it does a cross-sectional area at every individual point and plots the area in this form. So along this axis is the distance along the cord, and here is the cross-sectional area. So what we normally do is we flip it around. So okay, so just going back, so this scan takes about 7 minutes of scan time. If you had to add this on, but we can always do it on something that you are already scanning. And analysis takes about 10 minutes because it has to reformat all these sizes and calculate the edges. So we flip this plot around to show this is the length, and there are many features that you can see. One of them is the cervical enlargement right there. There is a dramatic transition in or reduction in cross-sectional area as we go from the cervical to the thoracic region, and there is a small region here that has a stable cross-sectional area from T4 to T9, and then that is the lumbosacral enlargement right there. And as I said, we plot it as a percentage distance, so we normalize for the length of the cord, so different patients with different height are not affected, and that means that these dotted lines, which indicate cervical and thoracic vertebral segments, fall very consistently. So we were happy to see, okay, so we've seen this plot, the same shape in many talks over yesterday and today. We can also go back to 1964 here, where they've done it on tissue. And they've seen that, I mean, this is the same shape, but although this is sizable diameter, this is very consistent with that. And I can also show you that they did not find any difference in age in the spinal cord. So I put the slide in. I did not have it before, but after yesterday's discussion, I put the slide in. Test request reliability. We find that 95% confidence interval is about 3 millimeters square, which is pretty good. So back to our natural history trials, we have 2 trials going on, one in HAM TSP, one in multiple sclerosis, and MRI is a major part of the trial. We have both brain and spinal cord MRI, and we have an extensive neurological examination. Some of these scores may be familiar, like EDSS, SNRS. We have time to walk, time to complete task of 9-hole peg test and 24-foot walk. And IPEG and Hauser are essentially done for in the HAM TSP trial. And then we have we also obtain CSF and blood from the patients and look for CD4, CD8 frequencies as well as proviral load in HAM TSP patients. So we have done this in 24 healthy volunteers, 17 asymptomatic carriers of HDLV, 45 HAM TSP patients, 40 primary progressive MS, 74 relapsing building MS, and 17 secondary progressive MS. Of course, none of these clinical scores match because the disease is very different. But this is what we have in healthy volunteers. So this is the average from the healthy control group. The thickness of the line is the standard error of mean. So that gives you an idea of what the variance is as well. And that's HAM TSP. And surprisingly, it is not just the T-spine that is thinner, but the whole spinal cord was thinner compared to the healthy volunteers. And asymptomatic carriers were not significantly different, or it almost overlapped on the healthy control subjects. So to do statistics, we chose 3 regions where the spinal cord was relatively area was relatively constant. So this is C23 region, the cervical enlargement C45 region, and the thoracic region, white matter region, maybe T4 to T9. And we see that indeed in all 3 regions, C23, C45, and T49, the HAM TSP's group average was much lower than the healthy volunteers and the asymptomatic carriers. To look for correlations, we did CD4 T cell frequency versus from the DSM versus C23, C45, and T49, and we find that there is significant correlations in each. And just to remind you that these are the cytotoxic T cells that are responsible for driving the atrophy. So we also looked at HDLV proviral loads, and the same thing. It's well correlated in all 3 levels. And with thinner cord having higher proviral loads. So we correlated with clinical measures of disability that we have, and we saw that disease duration, the higher the disease duration, the lower the spinal cord cross-sectional area. Similarly, for disability, higher disability corresponded with lower cross-sectional areas. But it was mainly in the T region rather than in the C region. So we wanted to take it one step further and compare individuals. So to understand what is happening in an individual, we had to we devised this method of plotting. In blue here is the average of healthy volunteers or asymptomatic group. The green here is the average of HAM TSP group, and this black is the individual's plot. And you can see that in this case, it is the T-spine is closer to the HAM TSP group, so we can make deductions like this. So we then divided these plots into 4 groups. This is an example. This is not an individual. These are separate individuals. So individuals that look like normal appearing cord. Individuals with C atrophic region defined by Z-score of less than -1.5. Individuals with T atrophy, and individuals with both C and T atrophy. And we find that in the HAM TSP group, not surprisingly, 78% of the subjects had C and T atrophy, 15% had just T atrophy, and 7% was normal appearing cord, which kind of makes sense. And looking at again, looking at differences between these groups, normal appearing cord subjects with normal appearing cord had much lower disease duration, or CTA had much higher disease duration, indicating that there's a longitudinal process going on here. And again, with the proviral load, the individuals with C and T atrophy had a higher proviral load. So we then went back and looked at longitudinal study of one person. So it's a longitudinal study. This person had no clinical progression in the duration of the study, and he had a disease onset of 8 years before the first time point here. So this time point is the first scan that we have, but this person had disease 8 years prior to that. So as you can see, so this is month 0, month 8, month 17, month 31. There is hardly any change in the spinal cord cross-section. It completely overlaps the cross-section area of the MTSP group. Nothing is changing here, which is a great control for us. And we can deduce that this person has mild or less progressive. So the second subject here is a showed rapid clinical progression. And so this is the first scan that we have of the person. And you can see that even in 2 months later, there is slight decrease in thoracic cord cross-section area right there. 12 months, you can see that, 12 months, you can see that the decrease is prominent. 15 months, it is quite dramatic here. And then you can see in 24 months, the C involvement as well. So it is definitely a progression of disease with, I guess, dying back phenomena right there. So again, this is a rapid progressive patient. Third case, the last case I'll show you. We have this patient, again, a rapid progressor, MTSP. Month 0, month 4, month 7. Now you can see it dropping down. The atrophy starting in the T region, and it's dropping down to the MTSP levels. Month 17, month 31. Now it's 17 and 31. Now it starts to affect the C regions. So again, another rapid course. So that is all about MTSP. I'll just talk briefly about MS. We can do the same thing in MS. We have 24 healthy volunteers here, secondary progressive subject 17 over here, and then the RRMS. And we can see that in relapsing remitting form of the disease, there is a decrease in the spinal cord, the cervical area, but not so much in the T-spine area. And surely enough, when we do the regional differences, we can see that this is a very busy graph, I apologize, but the healthy volunteers and relapsing remitting patients are different from the progressive forms of MS. But in the C, just in the C region, the healthy volunteers and relapsing remitting are also different from each other. Now there are 30 graphs if I to draw this, but so I just put in the table. The correlations clinical measures, I can tell you that RRMS showed a significant correlation with all the clinical measures that we measured, that we have. So it explaining about up to about 20% of variability in these clinical measures. SPMS, the correlation was much less, and in PPMS it was intermediate. So one thing that, so we're going back to the atrophy patterns. We're looking at normal appearing cord, cervical atrophy, thoracic atrophy, and cervical and thoracic atrophy. So if you just note that SPMS and PPMS clusters more or less have the same distribution of atrophy patterns, but in the RRMS versus SPMS, you can see that 75% of RRMS subjects had normal appearing cord, which changed to 28% in the SPMS. 12% had C atrophies, which changed to 31% in SPMS, and 11% had C and T atrophy, which changed to 39% in SPMS. So there is definitely progression again in this case, right? So again, we have 3 subjects. I just show you very briefly. Unfortunately, none of our subjects really converted from RRMS to SPMS, which would have been really cool to see. But the study is still ongoing, so I hope, well, not for the patients, but for the sake of studies, that somebody we can catch this at this point of conversion. But this person had no significant clinical progression. He presented at EDS of 1.5, then went to 0, and then went back to 1. So there's some clinical fluctuation in EDSS here. And you can see that there is very little change in spinal cord atrophy over 5 years. This person, again, no clinical, no significant progression, but what happened was in the 2-year scan, there's definitely change in the cervical spinal cord. So there is something going on here in the cervical cord, but not really showing up clinically, which means that it's probably in the brain. The damage already happened in the brain. This is inference. We don't know about this for sure. This is the third subject. Again, EDSS. So this EDSS change, his EDSS changed from 1 to 2 during the follow-up visit. But you can see here, time point 0, time point 29. There is definitely progression in the T and in the C regions. So there is atrophy sitting up there. But then after that, it's pretty stable. Okay, so final slides. We just thought that we should look at all the people. So since there is no progression or conversion from RRMS to SPMS in these patients, we thought we will just see, look at what is happening in subjects whose EDSS changed by 1 or increased by 1. So we have 14 such subjects in our study, and the follow-up interval was 1.6 years. And if we plot the EDSS versus regional SCSA, that is C23 region, C45 region, and T49 region, you can see clear trends. And this trend as a group is significantly different, larger than 0. So there is definitely a clear trend here in the C23 and C45 region, not so much in the T49 region. But of course, to avoid bias, we also looked at SNRS. So SNRS is reverse scale, so 100% is no disability, or 100 is no disability. And as the score decreases, disability increases. So, and you can see the clear trends here as well. And we just wanted to see, since it was, we are interested in thoracic spine, we want to see time to walk, for example, was correlated with this atrophy measures, and you can see maybe there's a non-linear relationship here. Our study is still going on, but it's very interesting to do this. So in summary, I want to say that this method seems to be a robust, reliable, and relevant marker for spinal cord atrophy, but there are some confounding variables, like, for example, the scanner effects we haven't explored completely. So the contribution to cord atrophy to disease progression depends very much on the disease. So it seems to be playing a more direct role in HAM-TSV than in MS, because we are not looking at the brain. Like somebody said this morning, we need to look at both together. Nevertheless, I think SCSA is a very good marker, and clinicians can benefit from it. And also that, so it's also relevant in, as an endpoint in clinical trials, maybe. And we can look at objective measures of our thinning with this, and it's correlated with various clinical and laboratory measures, and you can help select patients. I think this is a good point, because you don't want, if you're doing a trial or based on say cord thinning, you don't want to have somebody who's already got a thin cord, and or vice versa. So you want to you be able to select patients based off of this. So I think in general, what is important to note is that just one level, studying just one level of the spinal cord may not be sufficient, and doing the entire spinal cord gives you a better picture of what is going on. Well, thank you, and thank all those who did the work. I'll just talk about it. So C. Jacobson is the PI of the HAM-TSV protocol, and Danny Wright is the PI of the MS protocol, and Winston most of the programming for this. Sheila and Yair are clinical fellows in the lab. And thank you for your attention.
Any questions? Any comments? Hugo?
Yeah, very nice work. I was wondering about the MS patients that you showed and the changes over time. Were these patients under treatment? And
Yeah, so they're all generally on treatment. There's, we don't have any patient that is not treated. I want to say that maybe there are one or two, but they're all under various forms of treatment. So we haven't subgroup analyzed all of them.
And could there be something like Atrophy going on, explaining the discrepancy that you saw between?
Surely, yeah. Absolutely. And other thing to note is that inflammation per se, lesions can cause an increase in CARD. So that also needs to be resolved in MS, but in HAM-PSP it is very clear. Yes, sure.
Very interesting work. Do you do any kind of normalization? Spinal cord with highest question that was very difficult
Yes. So we have found that the only normalization that we've been doing this from 2013, I believe. The first study was published in 2013, and we have been doing consistently this on a clinical care basis as well. And I think the only normalization that needs to be really done is the length of the cord, because each patient has a different, the height is different. So the length of the cord is slightly different. So we want to normalize that. And then once we do that, the vertebral bodies generally fall very, very consistently. The dotted lines on vertical dotted lines were the vertebral bodies, and they fall very consistently. The other, so to take it one step further, I think, I mean, you can do volumes, but I think that again, you would have to, I don't know how you would want to. So instead of cross-section area, you could do volume of segment, but
we expect that people that are higher, that people who have a large spinal cord.
I haven't seen that.
It was discussed yesterday.
Yes.
It's also an open
right.
discussion because it's not known exactly how to do the right normalization.
Exactly, and I haven't seen that is what I'm trying to say. The only normalization that we have always done is just the length of the cord, and we just make it 100, right? So.
Any other questions?
Yes.
Interesting information is not looking at absolute difference between EDS scores, but maybe reaching the critical level.
Okay.
Because whereas
right.
there are some
yes.
critical milestones, EDS score
6. Yes. Yes, that's a good point. I think EDSS score, especially at the lower end, is a little bit subjective, probably. Maybe. I don't want to say that to this audience, but it's a little bit subjective. And also, the EDSS is very motor dominant, probably, and also there can be some other causes that can, So, for example, a relapse. Yeah. All right. Thanks.
Any other question? I have one question. Yeah. So you said that in HGLV myelitis, the thoracic cord is mainly involved,
yes.
right? But you found atrophy throughout the cord. How do you explain the involvement of the survivor and the bar?
Right. So once the axon, so it's the same cell that throws the axon from, and especially in the motor tract, the lateral columns is, I mean, it's there all over. But so what happens is that the axons die back. So this is, I think, it is a very good example to show, because we always talk about ALS and things like that, and we don't know if it is axon dying back or whether in degeneration. I think we can really see this in the individual case.
Okay. Thank you.
Thank you
again for your presentation. So we can move on to the last speaker, with Professor Frederick Bachhoff from the Center for Medical Imaging Computing, UCL. Okay, Professor Frederick Bachhoff doesn't need any introduction because everybody knows about his huge experience in MRI readers. We will talk today about spinal cord atrophy rates in randomized control trials.
Yeah, so welcome and thanks Farhan and Julien for inviting me. And the bad news is we're running late. The good news is that many slides that I'm showing have already been shown various times, so I will speed up a bit. No, on time.
We are on time. We're on time.
Okay. My disclosure. Chataway, which is a sympathetic trial. And here you can see that over time there was a clear advantage of the sympathetic group that had lower brain shrinkage than the untreated patients, which is a very highly significant finding. This trial, by the way, also the EDSS showed a positive finding. When you look at other outcomes such as cognition, you can't find it. So there's a gradient from seeing very strong effect to mild effect to no effect. If you go to more subtle measures. This one I already mentioned. I'm just showing the graph. This is the ocrelizumab study in PPMS. So this was in SPMS and this is CPMS, showing some decrease in brain atrophy, but there's still considerable atrophy over 2 years. Note that the rate of change is approximately 0.5% per year. So spinal cord, as I said, is important. We've heard that many times already. Again, illustrated in this illustration, where you can see a massive reduction in the cord size related to the EDSS cords, roughly. And the various ways to measure that. You know, I'm showing some data with MACA. But there are other outcomes that will yield similar sort of results. You can see a clear reduction in size, especially in the progressive groups. And you can also measure from brain scans, as Hugo has shown. So in this slide, if you don't have seen this point before. You can see that there's an overall decrease in the progressive groups, and you can see it progressing over time, but these are all cross-sectional measurements, and you can see there's quite a bit of variability at each time point. So there's a statistically significant effect on the group level, but on a single subject level, there's massive variation. And I think it's beautifully illustrated in this paper from the Basel group, which used yet another technique that I don't think we discussed. And they claim they measured the volume of the spinal cord, but in fact it's a volume over a fixed length. So effectively they're looking at the area, I think. That's the only thing that varies. But what is nice is that they show the individual measurements over many years. So they had the whole cohort together for up to 6 years. And whereas you can see that the relapse and remitting group have a higher starting point than the progressive ones, which is shown here. There's massive variability, which which just surpasses statistical threshold, but the variability over time is massive. And although you can see a trend for higher rates in the progressive group, it doesn't reach statistical significance just because of the enormous variability that occurs. And it's really good that it presents the data like this, because otherwise it's hidden. So this slide I don't think you've seen yet. Did anybody show this slide? No, it came with the invitation letter, you know. We have to invite you on one condition to show this slide. So it's really nice work. So this is moving the very popular registration-based technique from the brain to the spinal cord. Basically trying to segment the cord twice, finding the region where the two segments think the contour is, and then looking for the gradient along this contour to find the growth or the atrophy. And we've tested this with Marcello and Ferran in the Magnum's data set that you've heard about already. It's a longitudinal data set where we measure the cord twice, at baseline and at follow-up. So two separate measurements, and then you subtract it in your database and you calculate the percent volume change, also with a direct registration-based method. So in total, close to 300 cases and close to 100 controls, so a sizable data set. And here's the main results. So on the left, you see the cross-sectional area base change, and on the right, the GBSI. You can see that overall the trends are the same with patients, and I think it was over 1 year, Marcello, correct me if I'm wrong, having roughly 2% change, which is a lot, which is higher than we saw in the previous studies. Keep that in mind. So the trends are roughly the same with the two maps. The only thing that differs is that the variability reduces, which of course speaks in favor of the precision of the measurement technique, and also should be favorable if you want to do a clinical trial. Also, if you try to predict either the diagnosis or ESS progression, which was limited in this sample because it's only 1 year follow-up, you can see with the GBSI that you have a better discriminative value compared to the CSA-based methods. And then if you do the sample size calculation, you can see if you move from the CSA ones, which are the green ones, to the GBSI, which are the red ones. We should have inverted the colors, but anyway, it looks much better. But the devil is in the details, because even much better may mean it only means below 500 per arm, which is still a massive trial. And if you then look in the table for the small print, get sort of achievable sizes per arm, let's say in 100 or 200 per arm, which we would like for phase 2. You need a very potent drug to be, you could show an effect of a very potent drug, but if you have a more moderate effect size, and I think 30% is more the sort of effect that we see on brain atrophy outcomes, you still need massive numbers. So still very high numbers. And this is on relatively good image quality from Magnum centers, academic centers that all pay a lot of attention to image acquisition. So what we then subsequently did, we went back and looked at some existing data sets from real life trial data to see if we could replicate these findings and whether they stand up. So we went back to the INFORM study. So this was the fingolimab study, remember, which was negative on the primary outcome, was negative for brain atrophy, was positive for T2 lesions, also reportedly negative for spinal cord atrophy. So spinal cord imaging was acquired in approximately half of the patients. We went through the whole data again. It turned out that a lot of scans were extremely poor quality, despite the fact this was in a control set. So this was a data set acquired more than 10 years ago with 80 or 100, huge trial. With dedicated cord imaging, and these are examples of what you find in your typical scan. So infolding occurs, very massive shading, very low contrast in the images. So then we deemed only, let's say, about a third of the images were of what we would call good quality based on visual inspection. So there's a lot of scans, even with dedicated cord imaging, that are not of good quality. So here's a task for you, Julia, to work on this. Then, if we then compare the GBSA, you can see, as we saw in previous studies, that you can narrow the confidence interval and the rate of change. We noticed that the rate of change is on the order of 0.5 to 1% only, so lower than in the magnet data set that I showed you. And if you then do the power calculations in this data set, and let's focus only on the columns with the good image quality, which is this one and this one, you see again that you need massive sample sizes to detect realistic treatment effects. So let's say if you want to have a 30% treatment effect, you would have still hundreds. It's better with GBSA. You can see that only a third perhaps, but still the very high numbers. Here's another example of a trial which we reanalyzed, which is the Arpeggio Trial. This is using laquidimod, a drug that has now been discontinued by Teva, the development, but it's being used widely before. There would be various studies that were partially successful in relapsing-remitting MS, and some of these studies suggested that there may be more of a neuroprotective than an anti-inflammatory effect. Reason to trial this in primary progressive MS. And since it was a phase 2 study, we chose brain atrophy as the primary outcome measure, but we're interested in the spinal cord as well. So we decided to try to do also the additional spinal cord imaging. It's 1 year trial. And in the end, there were 181 patients with cord scans versus 283. So about 100 people missed the cord scan for one reason or another. And then there was, again, less than in the informed, but also here there was quite a few images with variable image contrast, to the extent the spinal cord toolbox failed to analyze and segment properly. So in the end, we had about 70% of data that was analyzable, but the actual number of scans available was larger if you took the brain scans and looked at the upper cervical part, which was included because it was a sagittal 3D T1. Here again, examples. Here we try to measure the noise ratio by this sigma, and you can see that. So these are examples of above and below the mean, and you can see it picks up the scans with a better image quality, better signal to noise ratio between the cord and the CSF. Again, this is from a clinical trial where we try to standardize the acquisition, but nevertheless, this is the sort of images that are being returned in a real-life clinical trial. And then we looked at various atrophy measurements with spinal cord toolbox. So we looked at the higher area, C1 to C2, and also C2 to C5, and then calculated with GBSI the rates of change over time at these two levels. And you can do similar things. Here's a bit of a busy plot. So we looked at C1, C2 with CSA and GBSI from the brain, same from the cord, and then from the cord scans only you can look at the C2, C5 with the two techniques. And the overall message is that if you use the registration-based method, then you get lower values, lower variability. Overall, relatively similar means, And again, as previously noticed, the rates of change are on the order of 1 to 1.5%. So overall, I think these trials show a bit lower atrophy rates than what the natural history studies suggest, and also a bit more variability than you would expect. And if we then do the power calculations, again, in the Arpeggio, so the orange ones are with the GBSI, they're favorable to compare to the CSA measurements. So that's the good news, but the bad news is that for 30% treatment effect, it's still in very, very high numbers. And again, you really need a potent drug to be able to show this. Whereas for brain atrophy, in similar types of real-life data, power calculations suggest that you can find a 30% treatment effect with approximately 125 per group. And that's been shown over and over and over again. So for some reason, despite the improvements in the measurement technique, we're not able to show these effects. So the conclusion of all this work was that, for a neuroprotective effect in progressive MS, we decided to stick with the brain atrophy, despite the fact that we were really enthusiastic about the spinal cord atrophy and spent a lot of time reanalyzing data, see if we could improve it. I don't think we're there yet. Although the registration did help. Interestingly, we saw also in these trials that the brain images are typically better quality and probably more suitable than the spinal cord. So the intrinsic image quality is better. There are fewer movement artifacts. So there's clearly work that can be done here. And beyond that, of course, there's a couple of other things that you would like to do. So there's some work ongoing here already in improving these acquisition techniques, perhaps not specifically focused on MS, because you may want to have also a sequence included for lesion detection, perhaps. We're not looking at the normal appearing brain tissue, of core tissue, which would require additional sequences, of course. We haven't looked at the lesions too much. As an outcome measure in core studies. And I think in the future you would perhaps like to develop some sort of composite measure and not, atrophy is a very crude end-stage type of phenomenon. So perhaps you would like to look at the composite of some intrinsic core damage, MTR or DTI, and the cord, which will then tell you ultimately what the total amount of damage in the cord is. And in closing, I'd like to thank some people. So the Magnum Study Group, Hugo, Marcello, Ferran, and the UK MS Society, who sponsored part of this work. Thank you for your attention.
Thanks, Frederick, for this interesting presentation. Questions?
So, Julien. So I have a very naive question about the. I was trying to interpret the power analysis. Mm-hmm. What do you call treatment effect? So like when we talk about 30% treatment effect?
Yeah. So ideally you do that versus a healthy control. So what is normal aging? So there's probably a little bit of normal aging and normal variability, and you compare to a untreated group, and then you simulate a reduction between the untreated and the controls.
So 30% means,
well, let's say if they have 0, just for argument's sake, and the MS patient of 100, you bring the rate of atrophy down to 66%.
Okay.
A lot of people do this by looking only at the MS group and then assume that the normal situation is 0 with 0 variability, which is not realistic. So it's better to have real control data where you know what the actual normal aging is and the variability of that.
And the rule of thumb in like, what do you usually see in terms of treatment efficiency? What are the like numbers?
Well, so I think for brain atrophy, I think 30% is sort of thing that you can see. If you look at other things, new T2 lesions in the brain, sometimes you see a 90% efficacy, complete suppression of new lesions. But for atrophy measurements, that's much more difficult to achieve.
Depends on the disease as well. This percentage maybe, Alzheimer is smaller, no? They look for 10, 20% of effect.
Yeah, well,
in brain atrophy or hippocampus atrophy.
Yes, please.
If we were about to use these spinal cord measures for clinical trials, do you think we should match our patients better to disease duration, because it seems that the rate of atrophy will change depending on when they were earlier into the disease and later, and then if this is not matched, then you will see the difference.
Yeah, well, yeah, you could match them on disease duration, or what would be the best is to match them on their atrophy at baseline. Because if you match for disease duration or EDSS, you still see this enormous variability. You saw that in the Basel study. There's enormous variability between subjects. And in fact, I bring this up many times, but people don't like the idea. Is, if your outcome measure is an imaging one, then I think you should include them also based on the imaging, because you want to homogenize your sample. That's very difficult in a clinical trial. Yeah. So I don't think there's a way around to reduce the variability between subjects.
When it comes to clinical application of atrophy measures from a clinical perspective, we are confronted with this very easy popping up first cross-sectional comparisons and also comparison over time under treatment. So what is, from an individual perspective, is your suggestion? Do we have to scan our patients before we start them on having treatment? Or to have their development over time? At least
you mean for their course?
Yes, because we can't do this actually, because everybody who's diagnosed with scan them or leave them without treatment. Right? So we still have cross-sectional comparison, and then later on, what is the percentage we need to expect to slow down the atrophy?
I have no clue. For an individual patient,
exactly, that's what we use.
Very difficult. Very difficult. I think we have very limited information that even for brain atrophy,
Nadia?
Sure about that. Sure.
Hi, thanks for the talk. One of the things that is always puzzling is the fact that we are advancing all these methodological methods of acquiring, microstructure of data and T and so on, and optimizing the acquisition part. But then when it comes to clinical trials, there's always the thing, you can't use something that has not been really validated a lot. But the validation, especially the sensitivity to a disease, comes also from large studies, which the trials are the best, probably the best platform to get them going. So why not pushing for at least adding one element of novelty to every trial that is happening? I know, but it's money in the long term. If then adding 10
but it's being tried.
It's
being tried. There are various studies. For example, if you go to the brain, have tried to use MTR, and it's a nightmare to get it standardized. And the failure rate is incredible.
In the past, but for example, in the cohort with the protocol that the huge potential has put, the community has produced. We have protocols that are now fairly easily implementable on almost any scanner. So at that point, it maybe comes down to the training of the technicians that need to put the slices in a certain way and agree to, to try to acquire the data in the best way possible. But I think that we are much more advanced today than even.
I agree. Yeah.
So maybe
we are, but it's tedious in real life. I'm fully supportive of them. We should try, or you could try it less in a subset. You say, with perhaps 20% of the subjects you try, because you need to have a certain volume. Because
But
to
I
give you an
think-
example for this platform study, you know, we're now being planned to enroll approximately 100 subjects, probably over more than 1,000 subjects, over probably more than 100 sites. 10 subjects on average per site, you know, which is not a lot. That means, you know, 1 a month. So you will see, you know, you instruct somebody, next scan, there's a new technician, can't find the protocol, makes a mistake. To get a routine and train people, it only makes sense to include a lot of subjects in a short period of time. And you know, with 100 sites or 100 plus sites, you have massive issues with variability in scanners, software versions, coils.
I know it's very difficult, but I think that if we don't find a way of getting over these, we will always
but I agree. It would be nice
on
to.
the 300 subjects, because if you do the power calculation on our diffusion data, you know, in theory, with advanced diffusion, some of the studies tells you that you have enough of 15 to 20 patients to see.
Yeah, well, I didn't show. So there's one paper that was shown yesterday was actually a UCL paper looking at primary progressive and suggesting that you could see these treatment effects with 20 people per arm. Well, that's obviously not generalizable to a normal multi-center clinical trial situation where you have much more noise and less progression also. You see a lot of these academic studies, they select a certain type of patients who are, you know, progressive and willing to participate, and then they measure all sorts of things. But if you just take the whole population, you often find less favorable, you know, smaller rates of change and bigger measurement error.
And to add to what Claudia is saying also is that we need to move to the more composite type of score, which is what you mentioned also.
Yeah.
Conclusion that we should not see each technique individually, but we win a lot of statistical power by combining them.
And I think, you know, combined brain and core examination is also an excellent way forward. You know, combined coils are becoming more and more available. Probably you immobilize the patient also better. So less movement effects, more homogeneous signal.
But also some of the scanners now allow you as well to use, I don't know how it's called because we don't have it, but I think the Siemens scanner definitely has the possibility of telling the scanner which particular brain or part of anatomy you want, and the slices are positioned based on what you tell them, right? That the user positioning the slices. So even that kind of error can be mitigated.
Oh, yeah, but that is
Auto align.
Yeah, that's available on almost all scanners.
I think that all the others have just use it.
That's it.
Okay, so last question.
Thank you. Is the pseudo atrophy also the issue in the spinal cord?
I'm not aware of it, but you know, I've not seen any reports on it, but I think it's conceivable, isn't it? But I've not seen any reports on it.
The second part of the question is now we have various treatments. Is the pseudo atrophy after treatment induction is it different in different treatments?
Yeah, in the brain, yes. Yeah.
Okay, thanks, Frederick, for your talk again. We can move on the conclusion.
Well, not too many conclusions or presentations for the conclusion. Just say thank you because we have had a very nice 2 days. We run on time, just 1 minute late or 2 minutes late. Now the people have some lunch box outside, grab one and have a nice flight home, or I don't know, by train, whatever. The people that is staying here in the afternoon for the spinal cord toolbox course is going to be another room inside UCL. which is in the building that is in front of the portico. We need to walk just down this street to the hospital, more or less, this tall white building, and then just down to the left, and you will arrive. It's almost 100 meters after coming to that. It's called Cruciform. It's red, the building is very easy to see.
Maybe we can all go as a group.
Yeah, well, up to the people. We have 1 hour and a half until the room is available for us. It's at 3 o'clock. Thank you very much. I hope to see you.
Thanks to Karen.
Thank you.
We need to